Good morning, everybody. I hope you, like me, enjoyed yesterday. Yes? This day will be at least as eventful and challenging and inspiring. So we look very much forward to a very nice program today too. The first speaker will be Dr. Leroy Hood, President of the Institute for Systems Biology in Seattle, in the United States. Dr. Hood graduated from the Johns Hopkins University School, that's in Baltimore, right? And that was in 1964, and he got an MD and from Caltech with a PhD in biochemistry in 1968. After 3 years as senior investigator at NIH, his academic career began at Caltech, where he and his colleagues developed the DNA gene sequencer and synthesizer and the protein synthesizer and sequences, 4 instruments that paved the way for the successful mapping and understanding of the human genome. Over his career, Dr. Hood has focused on diverse areas: molecular, immunology, neurobiology, cancer, neurodegeneration, genomics, proteomics, and systems biology. In recent years, Hood has pioneered systems medicine and a healthcare that is predictive, preventive, personalized, and participatory, the P4 program, P4 medicine. A pillar in biotechnology field, Dr. Hood has played a role in founding 15 biotechnology companies, including Amgen, Applied Biosystems, Integrated Diagnostics, and Arrival. He is a member of the National Academy of Sciences, the National Academy of Engineering, and the Institute of Medicine. Dr. Hood has co-authored numerous textbooks in biochemistry, immunology, molecular biology, and genetics, as well as a popular book on the Human Genome Project. The Code of Codes, and he has just finished up a text on systems biology. In addition to having received 17 honorary degrees from prestigious universities in the US and abroad, in 2013, Dr. Hood was presented the Medal of Science by President Obama. Leroy Hood is considered one of the most influential scientists of our time, and we now look very much forward to listen to him. Welcome.
I'd like to start with 2 points. One, about 15 years ago, a new approach to biology commenced called systems biology. It's global and holistic. I'll talk a little bit more about it later. But that systems biology approach has absolutely transformed medicine, giving us systems medicine and this thing we call P4 medicine. Considering P4 medicine, we've come to realize that something we've called scientific wellness is going to be in the future a fundamental part of the healthcare system, and it's a fundamental approach to dealing with the whole process of aging. And I think what's remarkable about scientific wellness is the idea that it will not only transform our personal health, but it will let us understand disease in ways we never could have conceived even a few years ago. So when I started at Caltech in 1970 as an assistant professor, it was clear to me that biology was enormously complex, as was disease at that time. It was also clear that we didn't begin to have the tools to deal with this complexity. And interestingly enough, we didn't have even a conceptual way of describing the complexity, and that later came to be called systems biology. And in fact, it reminded me at the time very much of the famous parable of the elephant and the 6 blind men, each feeling a different part of the elephant and declaring it's a spear or it's a stump or it's a fan. And that was very much what biology was at that time. People tended to look at one gene or one protein or one very simple system and not to see the biological system in the context of the whole that it was. And I was really intrigued with the idea to handle biological complexity. We needed to develop new technologies so we could create data to explore new dimensions of data space in living organisms and so forth. And in fact, I remember in 1973 reading this book by Thomas Kuhn, The Structure of Scientific Revolution, which talked about paradigm changes in physics. It made the really fascinating point, one, that paradigm changes are really difficult to initiate because they require that you think outside the box. And most of us never learn to think outside the box. It's really an important attribute to aspire to. And number 2, he made the point that even if you do succeed in thinking out of the box and creating a paradigm change, getting the community of scientists to accept it is even more difficult because most scientists are terribly conservative and they're very reluctant to give up what they learned earlier in their career. And I certainly found this to be true as I, over the course of my career, have participated in what I would call 6 paradigm changes. And you've heard about the first one. That is, at Caltech, we developed 4 instruments that basically allowed one to read and to write DNA. And later we developed several others that I'll talk about at a later point in time. What these instruments did that was really transformational for this whole issue of complexity was they allowed us for the first time to think about making high throughput measurements in biology, getting lots and lots of data. And of course, that eventually led to big data and analytics, a central theme in biology today. One of the instruments that we developed, the automated DNA sequencer, was what made possible the Human Genome Project, and in fact got me invited to the first meeting ever held on the Human Genome Project in the spring of 1985, where 12 scientists were invited to come and consider this possibility. And we came to 2 striking conclusions. One, that it was feasible, although technically extremely difficult at that time. But more interesting was the idea that the 12 of us that came there, supposedly sympathetic for the idea, were split 6-6 on whether it was a good idea. And those against it were fanatically against it because they saw it as the intrusion of big biology and the demise of small biology. And of course, the irony of that is the Human Genome Project, the first real big science project in biology, utterly transformed virtually every aspect of human biology. And it's absolutely facilitated beautifully small science, looking at things in much more detail and so forth. But for me, the essence of solving the human genome sequence was, of course, for the first time, one could think about doing proper systems biology because you had the knowledge of almost all the genes, and by inference, all of the proteins. Now, the automated sequencer to build really required the integration of engineering, of chemistry, of computer science, and of biology. And it was during that period of time that I realized that in the future, biology was going to be facing really hard problems, and it needed to live in a cross-disciplinary environment where the leading-edge biology could drive the development of relevant new technologies, and they in turn could create the analytic tools you needed to handle the data. And this cross-disciplinary vision I proposed at Caltech in the late 80s, the biologists utterly opposed it. And I'll leave it to you to think why they might oppose it. But Bill Gates made it possible to go to the University of Washington and start the first cross-disciplinary department in 1992. And it thrived there for 8 years. We invented the first major techniques in the newly emerging field of proteomics. We developed the software that fueled the whole genome project. I developed there the inkjet technology that allowed one to synthesize DNA arrays and enormous amounts of DNA, and on. This department was enormously successful, but unfortunately, the dean who hired me and promised me yet another floor after 4 years to create a systems biology institute died in the Himalayas in an avalanche, and the new dean came in with a totally different agenda. So after a few years in 2000, I resigned to start the Institute of Systems Biology, a nonprofit research institute that was dedicated, the first such institute, to essentially developing system science and its application both to biology and medicine. And that quickly led to these concepts we've already talked about of systems medicine and P4 medicine. We'll talk about those in considerably more detail. And I would say in many ways the final interesting paradigm change has occurred just in the past year when our institute affiliated with the U.S. third largest healthcare system to be able to bring the wonders of systems medicine directly to the patient's bedside. And what I'm going to do is talk a little bit about these last three paradigm changes. First, I'll talk about the Institute of Systems Biology now, with 9 professors and a $35 million a year budget and everything. But what the essence of systems biology is can best be discussed in terms of this Rube Goldberg cartoon. So in this cartoon, Rube Goldberg has put together 14 devices that, when appropriately connected, allow him to cool his soup. And the question is, what would you do if you wanted to take a systems approach to understanding that relatively complex gadget? And of course, the idea is, one, you'd want to define all the parts in the system. That's what the Genome Project did for us. 2, you'd like to understand what the individual parts did. 3, you want to know how the parts are connected one to another. 4, you'd like to know the dynamics of the device and how it moves with regard to cooling the soup. And then 5, you could begin to formulate hypotheses about how it carries out its soup cooling activity. And then 6, and this is a critical part people often forget in systems biology. Systems biology isn't fundamentally computational, it's fundamentally biological. So the ultimate objective is to perturb the system and formally prove that your hypotheses are actually correct. And that's really the essence of systems biology, only, of course, living organisms are infinitely more complicated molecular machines in this regard. Moving to the 5th paradigm change, of course, systems medicine and this so-called P4 medicine, predictive, preventive, personalized, and participatory. What I would say is systems thinking has provided 2 fundamental opportunities with regard to medicine. So number 1 is the idea that humans, like elephants, are complicated organisms. So we need to be able to generate an enormous amount of data for each individual to really understand what they're about. And we call this generation of data dense, dynamic, personal data clouds. And in the future, each one of you will have literally billions of data points that come from many different types of measurements and analyses. And the fundamental idea is that we are now learning how to integrate and model this data, so for each individual, we can identify actionable possibilities that will either increase your wellness or let you avoid disease. And we'll give you specific examples of this in a few minutes and everything. I think a really important part about this whole endeavor is this study, which we started in 2014. I'll talk to you about that in just a moment. Really was the first example of what precision medicine should be. Precision medicine isn't just about genome sequences and about clinical records. It is about making all of the other vital measurements that let you reconcile how your genetics and your environment come together to provide you with your health at that particular point in time. And that's what these dense dynamic personal data clouds do, and we'll talk in a little bit in a few minutes about what that's all about. So the first thing is generating these data clouds that are absolutely critical to understanding human biology and human medicine. The second thing that systems biology has brought to systems medicine is the idea of biological networks. These are the interactions of molecules and systems. They can occur at the level of the genome, at the level of molecules, at the level of cells, at the level of organs, or even they can occur at the level of organisms where you have social networks and the like. These are all seamlessly integrated together to, under normal conditions, mediate development, physiologic responses, and aging. And of course, if these networks become disease perturbed, they cause disease. So understanding how that disease perturbed network differs from its normal counterpart is the key to understanding the pathophysiology of disease. And it's critical to being able to develop early diagnostics and therapeutic approaches toward disease and everything. Now, I'm going to talk about one experimental system that we've studied for the last 10 or 12 years in enormous detail, because it is a beautiful example of what systems thinking is going to be doing for humans in the future. And this is a form of neurodegeneration in mice that's caused by injecting into their brains activated prion particles. And these activated prion particles cause a neurodegeneration that in mice generally takes of the order of 22 weeks. And what we did with these mice was to sample different individuals at different time points along the 22-week period by analyzing the RNA molecules that were present in their brain. These are called the transcriptomes. And we compared the transcriptomes from the disease mice against the transcriptomes from their normal mice to be able to see which genes changed in the disease process. And to our horror, we found that almost 7,400 genes changed. And it turns out most of those are biological noise, and we developed really beautiful ways for subtracting that biological noise away. And we ended up with a conclusion only about 300 genes really dictated this neurodegenerative process. At the same time, we looked histologically at the brains of the animal across this 22-week period. And what we found was that there were 4 major processes that were involved in this disease. Something called prion replication, accumulation, glial activation, and then 2 forms of neurodegeneration. There were biological networks, at least partial networks, that represented each of these 4 systems, and we could map beautifully the 200 of the 300 genes into these 4 major systems and the other 100 actually identified 6 smaller processes that had smaller networks. And the 10 networks together, and looking at their dynamics across time, explain virtually every aspect of the pathophysiology of this disease. But the really important take-home lesson was that the networks became disease-perturbed in a sequential manner, starting first with prion replication and accumulation, and then glial activation, and then the 2 forms of neurodegeneration. The reason this is important is in studying human disease, it's essential to know what the very first event is in the transition from wellness to disease. Understanding at that time point the disease-perturbed networks gives us the possibility of early diagnosis and eventually of being able to reverse the disease at the earliest stage. And the reason this is very important is that we're going to talk later about how we can now do for the first time exactly the same kind of studies in humans. But there is one more important point, and that is, in order to know where you are in this neurodegenerative process, we had to actually sample, and in doing so killed, the individual mice at different stages of this disease process. And if we're going to do it for humans, we can't do that. So what we decided was to test the proposition that these disease-perturbed networks would actually create changes in proteins in the blood that could reflect this disease initiation and progression process. So what we did was to develop a series of techniques which I won't describe that allowed us to identify in humans 100 proteins in the blood that are relatively brain specific. They map back to many of the major networks we know about in the brain. And of course, in a normal individual, excuse me, those 100 proteins will have one set of levels. But if any one of those networks becomes disease-perturbed, that alters the level of its cognate proteins, and that'll be reflected by changes in concentration in the blood. So we can distinguish healthy brains from diseased brains, and because each different disease perturbs different combinations of networks, we can distinguish what type of disease it is. We found that there were a number of these brain-specific proteins that map to the 4 major networks in the mouse. And we were actually able to use mass spectrometry, measuring the concentration of these 15 proteins that mapped into each of the 4 major networks, to show that they too, as with the transcripts, were successively disease-perturbed in precisely the same order, and they only differed by a few days from the initial events that we were able to prick out in the brain. So it means we've made the blood a window into distinguishing wellness from disease and following the progression of disease. And we'll talk more about that later with humans. So by 2006 or so, we had clearly defined systems medicine and P4 medicine. And in many ways, we were at an enormous point of opportunity, but we needed to develop the technologies and strategies to fundamentally change medicine. And we had many ideas. We put many grants into the National Institutes of Health, most of which failed because they asked that everything be half done before you do it. But we were very fortunate in 2000. I was very fortunate in 2007 to meet the Minister of Finance of the state of Luxembourg, who was a dominant political leader at that time and had decided he was going to transform the economy of Luxembourg from a 90% dependence on financial services to bring in healthcare and biotechnology. So he asked me, would I write a proposal that would help them do that? And we wrote a proposal where we built an institute that mimicked our own at the University of Luxembourg that's been enormously successful. And on the other hand, what they did for us was to give us of the order of $100 million over a period of 5 years to develop the technologies and strategies of systems biology. And these enable us to take on daring, high-risk projects, and almost all of them ended up being successful. And we'll show you some examples of that in just a moment. But it put us at an enormously interesting tipping point because we could really do fascinating kinds of things now. And so in 2012, I made the proposal that the way to bring P4 medicine to the healthcare system was to engage 100,000 normal individuals and study their wellness and optimize their wellness using the technologies that we developed and so forth. And in 2014, we did the first 108 of these individuals, and I'll talk about that in just a few moments. What also happened, of course, was the next year President Obama proposed precision medicine and doing essentially exactly the same dense, dynamic, personalized data clouds that we'd suggested for scientific wellness. And of course, again, with the affiliation with the Providence Healthcare System and ISB, it made it possible to translate all of these ideas directly to the bedside, and we'll tell you where we stand before. So here are some of the technologies that we developed in the course of the Luxembourg Agreement. And I'm not going to go through and talk about all these, but I'll talk about two that I think are really important. One, third-generation DNA sequencing is the ability to sequence single DNA molecules and to do the base detection electronically. And what this gives you is the possibility of very long reads, of easy automation of the front-end part of the DNA sequencing. And it's my guess in a 5 to 8 year period, we'll have third-generation sequencing, and human genomes will cost less than $100 a piece rather than $1,500 a piece today. A second area that ISB really pioneered is that of proteomics, where we developed, together with our initial co-founder at ISB, Rudy Ebersol, and later in collaboration with him, number one, targeted proteomics, which lets us look at 100 proteins and very complex mixtures quantitatively, and I'll show you an example of that. And number two, we've developed, helped develop a new type of proteomics called swath proteomics that will let us be global and measure at one time quantitatively 6,000 or more proteins from, again, complex mixtures. Now, the strategies that we've developed in many ways are even more powerful, and you can see several examples of those listed there. And I'll just mention two. One, making blood a window into health and disease, and I'll show you a beautiful example of that in just a moment. And then two, these dense, dynamic, personalized data clouds, which I think will absolutely transform medicine. So, making blood a window into health and disease, I'll show you how we've attacked a problem in lung cancer and solved it by using a systems approach to creating diagnostic biomarkers. So the essential problem is that in the US there are 3 million nodules in the lung that are seen a year, and that poses for the pulmonary oncologist the question of what do you do with those nodules? Are they lung cancer? 600,000 of these go to surgery. These are costly surgeries. And more than half of those that go to surgery are done on benign nodules that increase morbidity and increase cost to the healthcare system. So our objective was to develop an assay that could distinguish a benign nodule from a cancerous nodule. And we approached this by a systems approach to diagnostics. We started with 400 proteins roughly, and we did a whole series of filters that allowed us to go down to 13 that did quite a good job at distinguishing benign from neoplastic nodules. And we actually moved to 2 of those that did a superb job at doing this. So the 2 markers allowed us to identify more than 50% of the benign nodules at a 95% confidence. And what this obviously allowed us to do was to save the healthcare system roughly $4.5 billion a year just by ruling out almost 40% of the surgeries that were necessary. Of course, this improved the quality of life for people with lung cancer, knowing that your nodule was or was not neoplastic. And what was really interesting is after all of these filters, which I haven't described, that got us from 400 to 13 proteins, you can say, what exactly did those 13 proteins represent? And it was striking that 12 of the 13 mapped into 1 of 3 disease-perturbed networks in small cell lung cancer. So the filtering system reflected the biological process of lung cancer very ably. And this is really an important slide that I really want to push. Freeman Dyson is really a terrific writer. You ought to read him if you ever get a chance. His point, new directions in science are launched by new tools and strategies much more often than by new concepts. The effect of a concept-driven revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that need yet to be explained. And the Human Genome Project was absolutely a beautiful example of that. So by 2013 or so, we saw then a convergence of systems medicine with the new digital analytics for self-measurements, with big data and its analytics, and with social networks. And these allowed us to articulate much more clearly just exactly what P4 medicine should be. And to show you how different P4 medicine is from contemporary medicine, let me stress the 6 major differences. So 1 is it's proactive rather than reactive. 2 is it focuses on the individual and not on populations of individuals. 3, it's a major focus on scientific wellness as well as disease, and not just on disease. And of course, 4, it's all about creating these dense, dynamic, personal data clouds that are so critical for wellness and understanding disease. And then 5, it throws enormous skeptical view on the classic way of doing drug trials, which is to take 20,000 individuals and give them a cancer drug or a placebo and extract from that population responses and deduce how well the drug is done. And of course, the fault of that is each of those 20,000 individuals is unique genetically and unique environmentally, and you have no business averaging them. In fact, what the averaging does is increase the signal-to-noise kind of ratio. So in P4 medicine, what we'll do is we'll have 20,000 individuals, each with their dense dynamic personal data clouds, and we'll stratify those individuals based on their own unique properties according to features you're interested in, response or failure to response to a drug and things like that. And to stress this, I'm going to show you in just a moment how poorly the classic system for drug discovery does. And finally, I'll just mention that social networks are going to be key in this new world, both for learning about P4 medicine, for crowdsourcing, and ultimately for advocacy for changing the system. The healthcare system is every bit as conservative as the scientific system and resists every bit as vigorously dramatic new changes and everything. Now, here's really a striking slide. These are the 10 most sold drugs in the US, and the orange individual indicates how many people respond to the drug in an appropriate population. So the numbers vary from 1 in 4 to 1 in 25. And that means almost all the people that get the drug are wasting the money, and they're exposing themselves to the complexities of cross-reactivity and bad reactions to the drug. And this comes from doing population studies where you can't distinguish the orange people from anybody else. But with the P4 approach, we'll be able to pick the orange people out immediately and to assign the features that will in the future allow us to distinguish for a given drug who's orange and who isn't. And then you can give the drugs to appropriate kind of people. So for P4 medicine, two major ideas: scientific wellness and understanding disease and so forth. And what I would say is, and I'll define what scientific wellness is in just a second. My own personal view is that scientific wellness will create a whole new thrust in the healthcare industry. And I think within 10 to 15 years, its market cap will far exceed that of the old-fashioned disease industry or current healthcare industry. And the really key lesson to take home at this point is exactly what these dense, dynamic, personalized data clouds can do. So one, we can interrogate each of you to identify the actionable possibilities that will allow you to optimize your wellness. And we'll show you some examples of this in just a few moments. Number two, and this is really an important point, as we follow these people longitudinally with these dense dynamic personal data clouds, we'll begin to see wellness to disease transitions for every single common disease. We'll be able to identify the initial disease-perturbed networks, to develop the early diagnostics, and ultimately the therapeutics that will allow you to reverse a disease before there's ever a phenotypic symptom of the disease. In a sense, this reversal will be the preventive medicine of the 21st century. We'll be able to diagnose the very earliest stage for most diseases and reverse it. And of course, these dense dynamic data clouds will let you study disease in a way we've never been able to study disease before. Looking at the initial events, if you can guess who is going to get the disease, but following progression, following the response to therapy, and hopefully following the response back to normalcy and everything. And finally, I'll just say that in the future, individual N-of-1 experiments are utterly going to be the key to deciphering many of the complexities of medicine that we can't even touch today. And a classic area that I think will revolutionize is nutrition. And I think, frankly, nutrition is in the dark ages today. And for the first time, we have the tools to look at it in very, very different ways. So why is scientific wellness so important? Well, one is chronic disease is really skyrocketing in our populations. Number two, we're aging. All of our populations are aging in striking ways. And to give you an example of that, you can do a calculation that says 50% of the children born in this calendar year will live to be 100. So the question is, what's the quality of their life for the last 30 or so years of living to be 100? And we want it to be absolutely superb. And I think another striking fact, when a recent study was done on the determinants of health, one was able to demonstrate that genetics contributed 30%, that the environment and behavior contributed 60%, and the healthcare system only contributed 10%. And of course, the key thing about these dense, dynamic personal data clouds is they are the only way that we can use to assess how your genetics are clashing with your environment at a given point in time. And that's why they are so critical. So in 2014, I persuaded 108 of my friends to go through a year-long study with these dense dynamic personalized data clouds. 50% of them went into the study really skeptical. So remember that statistic. And what exactly are the measurements we made? First, we examined the complete genome sequence, all the genetic variants for each individual. Then every 3 months, we took blood and saliva to do clinical chemistries, metabolites, and proteins, and we carefully selected those. And then again, every 3 months, we took stool samples so we could do the gut microbiome. The microbes that live in your gut have an enormous impact on your health. We're just beginning to learn about it, and this is going to be a great discovery study. And then finally, we've given our subjects Fitbits so that we can record activity, quality of sleep, and pulse rate, and other things like that. And basically then what we did is every 3 months we took the blood samples for analysis as described above here. And a really important point is once a month, every individual was called by a coach who described for them the actionable possibilities, both defining what that meant in simple terms and putting it in the context of their own healthcare objectives, which they'd learned at the very beginning of the study. And these health coaches are absolutely critical for integrating and analyzing these data, taking the actionable possibilities to the individuals and persuading them to act on it. And in fact, the health coaches were responsible for more than 70% responses to actionable possibilities, really a much higher number than I ever suspected we would be able to get. What was also remarkable was the data itself, and I can only give you the briefest of thumbnail sketches. But to make an analogy, I think these dense dynamic individual data clouds are exactly like the Hubble telescope. It, for the first time, allowed one to look at the universe with a resolution never before achieved. And so these dense dynamic personal data clouds have allowed us to look into human biology, into human disease in ways we could never imagine. And to give you one example, we were able to take individual data bits from each of the 6 data types and do several types of statistical analyses to show data bits in one type that correlated with data bits in the other type, and we found almost 5,000 of these correlations. These correlations give us new insights into biology. They tell us systems that are interlinked that we never knew were interlinked before. They give us new actionable possibilities. They've given us some really striking hints about good biomarkers and possible therapeutic targets. So there's enough small science in this chart probably to put 1,000 people to work for years and years. So there are enormous opportunities represented here. Now, the other thing we learned is from the genome sequence, we could, for 60 or 70 GWAS diseases, these are genome-wide association study diseases, we could actually determine the genetic risk for the individual. And that's because these GWAS data were cast in terms of probability, so we could go through the entire genome and look at all the variants associated with the disease and add up their probabilities and translate that into potential genetic risk. And what we've been able to show with 5 different types of disease now is that the genetic risk increasing from left to right correlates beautifully with the disease phenotype in these patients. This is LDL cholesterol, the bad cholesterol, and you can see a beautiful correlation from low risk to very high risk. And we can do this for thousands of metabolites and proteins and so forth, and they give us all sorts of striking new diagnostic and therapeutic kinds of correlations. Here are some of the diseases that we'll be able to do genetic risks for. And I'll talk later about Alzheimer's because we're going to do really interesting experiments with people that have a very high risk for Alzheimer's disease. Another thing we could look at are transitions from well to greater wellness. And we use the metrics that changed in that transition to begin fashioning a metric for wellness. And we think we'll be able to develop metrics both for physiologic and sociologic wellness. We were also able to look at people that went from well to less well, and a few did. We were able to look at people that went from wellness to a disease transition. And again, learning that earliest transition and reversing it is the key to preventive medicine. And finally, we were able to follow the progression of disease, the response to therapy, and ultimately, hopefully, the return to health and so forth. And finally, we had 5% of the individuals had chronic diseases, and for a number of these, we were able to see a reversal back to wellness. We had several with hemochromatosis where we had striking reversals once we managed the disease phenotype appropriately. But each of the individuals, when they did something actionable, could look at their chemistries and see things were improving, and this was an enormously positive reinforcing event. When we looked in the initial clinical studies at the individuals, 91% were nutritionally deficient, some in really serious ways. 68% had inflammatory responses. They could either be diet or they could be infections. And we saw a lot of both and reversed a lot of those. We had 59% with cardiovascular complications and 56% with prediabetes. 53 out of 108 were pre-diabetic. We moved 7 of those to normal, and we moved the other ones over strikingly toward the more normal kind of stage. And finally, genetics, your genome tells you a lot about how to diet, how to exercise. There are 300 some variants that predict your susceptibility to various types of athletic injuries and so forth. We actually had about 4 people that had mercury toxicity. 3 of the 4 were due to eating excessive amounts of tuna sushi. When they went off that, the mercury reversed down to normal right away. And the 4th one was amalgam fillings that were replaced. And again, it went down to normal. So you can see then that we had striking correlations. We could do genetic susceptibility, and that opens up lots of exciting things. And then we can look at these state transitions, which themselves are absolutely fascinating. So what exactly did the pioneers think? 108 out of 108 thought it was the experience of a lifetime, and it really had changed it. And what was remarkable is everybody stayed on. We had no one dropping out. And so that, I think, is testimony to the power of what happened. But the first insight was most of them were excited because given data, you can actually make your own health determinations. You didn't have to have a doctor telling you what to do. They were also excited because they came to understand your genetics don't determine your destiny, rather your potential. And there are a lot of ways to avoid many of the genetic limitations most of us have. Not all, but most. And then finally, they were really struck with the fact that most people thinking themselves well, I think would have been in the bottom quarter of wellness, and that with the actionable possibilities, they really elevated themselves with regard to wellness and so forth. So at the end of 2014, we decided to set up a company called Aravale, whose mission was to bring scientific wellness to consumers. And it has been a spectacular success, almost without advertising. It's gotten 1,400 customers in the first 9 months or so. This is in the Seattle area, and we're beginning now to bring this to California, and we'll move to other states as well. And the business plan is really twofold. One is to optimize consumer wellness and help them avoid disease. And of course, the second is to create a longitudinal database that will itself transform the healthcare industry. And I'll say a word or two more about that in just a moment. Now, ISB and its affiliation with Providence that I spoke is really going to be very much focused on these dense dynamic data clouds and using them all the ways we've talked about. We are also really developing new assays that will bring the cost of the assays down, because that's the limiting factor in scientific wellness. It's now about $5,000 per person per year, and that's way too much. But we're on a Moore's law decline on the assay costs and so forth, and my prediction is in a 5-year period it'll be 10% of what they are today and so forth. And we're also developing a lot of really striking new analytics for being able to deal with all of these different types of data. So again, this affiliation I mentioned that occurred back in April has really transformed what ISB can hope to do. And our partner, Providence, is really a striking partner. So it's in 7 states, it has 50 hospitals, it's got of the order of 10,000 or so physicians, and it has access to more than 30 million electronic medical records, and we're developing now systems for beginning to mine the records for all of the obvious things that one can begin to think about. So it represents an unusual opportunity to look at data that already exists, but even more, it has given us the opportunity to propose 4 pillar projects, and I'll describe each of those very briefly. So the first is scientific wellness. Providence is going to take 2,000 of its employees and put it in the Airvale program for a 3-year period, and we'll compare it against 2,000 employees that are only given normal wellness treatment. And this 3-year study will do 2 important things: unequivocally demonstrate the power of scientific wellness in transforming people's lives, but even equally important, it'll give us the economics for understanding just how much scientific wellness saves the healthcare system. And it's going to be, I'll guarantee you, a staggering sum of money. The second pillar that we're looking at is taking 200 patients newly diagnosed with breast cancer, putting them on the dense dynamic personalized data cloud regimen, and then following them through treatment to post-treatment and bringing them back to wellness as quickly as we possibly can, with the wellness being defined in part by the initial studies before treatment and so forth. And this will be done again against a control group, and you can do this for any type of cancer, quite obviously. The third area that we're really excited about is Alzheimer's. We're collaborating with Dale Breseden at UCLA, who's developed a 36-point regimen that looks in very preliminary studies like it can fantastically reverse the earliest stage of Alzheimer's, bringing people back to normal and maintaining them at normal as long as they stay on the regimen. So we think with appropriate experimental and controls, we'll be able to demonstrate the power of this regimen for reversing the early stages of Alzheimer's. And the second project we're going to do is take 200 individuals at very high genetic risk for Alzheimer's, and we'll start them on this program and demonstrate that they never transition over into Alzheimer's. So we think we can do both reversal and prevention. I'd be glad to talk in more detail about how we're going to do that in any time. And then the fourth pillar is to study a disease now totally intractable, glioblastoma. Usually people die within 18 months or so. And we're going to really focus on two new kinds of opportunities. One is immunotherapy, and that's a real challenge to this tumor because it has a low mutational burden. And in general, tumors that have low mutational burden aren't good candidates for immunotherapy, but we have ways to think about it with new approaches. And number two, even more exciting to me, is I think cancer is exactly like AIDS. It should be treated from day one with triple drug therapy. So glioblastoma is really nice because from the resected tumors, in every case, we can get beautiful tumor stem cells that can be expanded, and we can use those in microtiter plates to scan for their susceptibility to drugs for the individual, and then later their susceptibility to combinations of drugs for the individual, and that's the strategy that we're going to be employing. So in closing, let me just talk about what I think the future of healthcare is going to be. One, P4 medicine has really come to healthcare. And two, precision medicine is going to be absolutely a central feature. These dense, dynamic, personalized data clouds will let us assess genetics and the environment. Three, we'll be able to optimize wellness for individuals, as I discussed with you. And four, we'll be able to use these individuals to get, for all common diseases, their earliest transitions and learn to reverse them. A new type of preventive disease and so forth. I think what is also going to be true is that scientific wellness needs to be a lifetime journey. And so let me make 2 points. Eric Topol has studied a group of 1,000 individuals at the Scripps Institute that are 90 years or older, never been sick a day in their life, never taken a drug. They're in their 90s, fully capable. He calls those the elderly. So that's point one. Point number two is I learned when we were studying 18 individuals, 115 or older, and we were looking for genetic variants that predisposed to aging, and the number was not large enough to draw any striking conclusions. But what I did learn is when you get to be 100, almost all people die very quickly from a complete systems failure. So my hope for scientific wellness is that if people ascribe to it for a lifetime, carrying out all of the new emerging actionable possibilities, that we can elevate ordinary people to the status of the elderly, going into the 90s fully functional and capacitated and everything. And of course, all we have to do is get you up to 100, and then you can deal with the total systems failure after that. So anyway, I think that healthcare is going to have its costs dramatically reduced by this scientific wellness. I think we're going to create a new wellness industry. There'll be many companies that will use these dense dynamic data clouds pointed at certain diseases to bring wellness to people before they get the disease and so forth. I think the data will transform how pharma, biotech, nutrition companies, and diagnostic companies practice their art. We can, for the first time, let them deal with signal-to-noise problems they didn't even know existed before. And of course, finally, my dream is in a 10-year period, we will use Moore's Law to bring the cost of scientific wellness to a place where we can take it to developing countries as well as underdeveloped countries, and even begin to think about the democratization of healthcare, a dream that was inconceivable even a few years ago. Lots of people do these things. I won't go through and mention any of the names, but thank you very much for your attention.
Thank you, Leroy. He's leaving the stage, realizing that there is no time for questions, but there will be ample time for questions at the discussion session just after the lunch. So please write down what you want to ask, and we will have a very nice discussion then. I thank Leroy once more. Fantastic talk.
Thank you.
Good morning, everybody. It is my great pleasure to introduce our next speaker, our dear colleague and friend, and Jubilee Professor at Chalmers this year, Professor Walter Longo. Professor Longo is Edna Jones Professor of Gerontology and Biological Sciences and Director of the Longevity Institute at the University of Southern California at the Leonard Davis School of Gerontology in Los Angeles. Dr. Longo studied biochemistry and jazz performance, and he plays the guitar, by the way, and received his biochemistry PhD from UCLA. Dr. Longo is also the director of the Longevity and Cancer Program at the IFOM Institute of Molecular Oncology in Milan, in Italy. His study focuses on the fundamental mechanisms of aging in simple organisms and mice, and also how that can be translated to humans. Longo Laboratory has identified some of the key genetic pathways that regulate aging in simple organisms and was able to demonstrate their inactivation can reduce the incidence of progression of multiple diseases in mice and humans. His lab has described dietary and genetic interventions that protect normal cells but synthesize cancer cells, and this has been tested now in several places on human cases. Dr. Longo received in 2010 the Nathan Schock Lecture Award from the National Institutes of Aging, and in 2013 he received Vincent Cristofalo Rising Star Award in Aging Research from the American Federation of Aging Research. Welcome, Walter.
Okay, thank you very much. So I'll tell you essentially about two things that we're doing in the lab. We've been doing actually for a long time, and one has to do with the genetics of aging, and then the second part has to do with how do we intervene in ways that are really trying to get this to work in the near future or even now. So I'll show you in some cases where we're running clinical trials, and there are actually food programs that we're utilizing to improve health. And so you've seen about Madame Calmont from several speakers, and the point of showing Madame Calmont again is that she obviously, as Linda Partridge pointed out, she was obviously genetically protected, or most likely genetically protected, and we also know this from Emma Morano, the Italian lady who's now almost 117 years old. And she eats 3 eggs a day and meat every day. But then when you look at her family, almost everybody made it to, everybody made it to 90 years of age. Several people in her family made it to over 100 years of age. So we know that this is very, very rare. And so they're genetically protected. And this is how most likely they get to these extreme ages. And so when we first started a long time ago in the early 90s, some of us, including myself, first thought about doing this in mice and humans. So we started trying to understand aging in complex systems, but that was very, very difficult, and we realized it right away. So a group of us, and you heard several talks at this conference, say this is not going to work. If we keep trying to look at these very complex systems and trying to understand how they age, we're not going to be able to do it. So the gamble was, let's go to a simple system. In my case, was yeast, Saccharomyces cerevisiae. You heard about that also from Thomas Nyström's and other. So we'll go to a simple system and then hope that A, we figure out how the simple system ages, and I'll show you about the genetics first. And then the B, that actually understanding how the simple systems ages is going to tell us how the complex system, the more complex system ages. So we actually, this is 20 years of work by my laboratory, really using all kinds of techniques, some of these beautiful techniques like transposon mutagenesis that were available back in those days in the early 90s for yeast. And those and other techniques allowed us to really understand and identify this network. I mean, this is our work on aging, but obviously there are many, many scientists that contributed to understanding of these systems at just the genetics and molecular level. So we had really tremendous help from hundreds of laboratories. But here, the summary of this. First of all, I developed something called chronological lifespan. I wasn't satisfied with the system available at the time called replicative lifespan. And so I developed a yeast chronological lifespan method that basically allows us to follow the aging of yeast as yeast ages in the wild, right? And figs, rotten figs and rotten grapes. So using this chronological aging system, we were able to identify two major pathways. One is what I call the sugar pathway, which activates RAS, adenylate cyclase, PKA. And when this is active, this RIN15 serine threonine kinase actually is inactive, and this contributes to keeping these three transcription factors, MSN2, MSN4, GIS1, they're zinc finger transcription factors, also inactive. And the other pathway is what I call the protein pathway, the amino acid pathway, particularly in yeast is serine, threonine, valine, but in mammals it's likely to be a little bit different. And these amino acids control the PDK, PKH pathway, and the TOR pathway, and they converge on this S-cis kinase called SCH9. And this SCH9 seems to be really central in the lifespan regulation, so in accelerating aging. So you have a lot of sugar, you have a lot of amino acids. Together, they activate this network, and this network shuts down these protective transcription factors. You heard about these phosphotranscription factors, and really these are the equivalent of these phosphotranscription factors that you heard about yesterday, these protective factors that regulate many genes involved in aging. So if you either remove glucose, and I'll talk about that in the next section, or have mutations in this pathway, you get lifespan extension. For example, if you delete RAS gene, the yeast chronologically lives twice as long. And if you delete this SCH9 that I told you about, now you get about a 3-fold lifespan extension. And actually, if you delete both, you can get up to 10-fold lifespan extension. So these yeasts are completely, the lifespan of the simple organism is completely reprogrammed, and in allowing it to live as much as 10 times longer. And so I told you there was a gamble, and I think it paid out. Why? Because, for example, the TOR pathway that I just told you about, S-cis kinase, now it turns out to be very much at the center of aging in most of the model organisms that have been studied. And also, if you see here, this is the SCH9 mutant, so the mutant for which the longest lifespan extension has been obtained. And these are the flies that Linda Pardage studies, the Chico flies and the IGF-1, insulin deficient flies, and they also have record longevity extension. And these are the mice that John Kapczyk and Andrei Barkey studied, the dwarf mice, and they have record longevity extension, right? So this is probably not coincidental, and this tells us, and these are in fact are all different genes, but they seem to be genes all involved in the growth of these organisms. And but as we started moving from yeast all the way to mice and then humans, obviously these mice were very interesting to us, and these mice, as I mentioned, they lack either growth hormone or the receptor for this growth hormone, the growth hormone receptor. And I think the beautiful work by Andrew Barkey and John Kapczyk led to not just the identification of these mice that can live 40% longer than normal, and even twice as long as normal when calorie restricted, right? This is really unbelievable if you think about making a mouse live twice as long just by a mutation and a dietary intervention. But more importantly, half of these mice make it to the end of life with no pathology, right? They don't get sick, so they live 40% longer and they get sick a lot less. Consider that the normal mice, 90% of them will develop some pathology, similarly to human beings, right? So this is really incredible if you think about this idea of don't focus on diseases, don't even focus on disease prevention, disease by disease, focus on aging. Why? A mouse lives 2.5 years and starts developing cancer at 1.5 years of age, right? But there is no human being, or very, very rarely, that develop cancer at 1.5 years of age, right? So the answer really is all there. It's all about aging. And if you can not necessarily do anything dramatic to aging, but just even relatively limited effects on the aging process can have dramatic changes in the lifespan and also in the health span, so the disease incidence in these organisms. So we knew about the super dwarf yeast and the dwarf flies and the dwarf mice, and obviously our question was, well, what about the people? Yeah, if this is true, it is all conserved, then if we find the people that have this same mutation as or similar mutation, or let's say analogous mutations, then these people should live longer and should be much healthier. And now, luckily there were 100 of them, all of them followed by Jaime Guevara down in Ecuador. So as soon as I heard about that over 10 years ago, we started studying this group of people, and they, like the mice, lacked the growth hormone receptor. And so our question was, do we see any difference in diseases? And also, the concern was that they did not have a healthy lifestyle at all. So they drank a lot, they smoked, they ate terribly. And so we were a little bit worried that maybe we picked the wrong group and that we were not going to see much. But it turns out that, as obvious from their behavior, that actually this is the growth hormone receptor deficient population, and their obesity is much higher than the control population, right? So not a good start until you look actually at their glucose level, which are normal, and eventually we did glucose tolerance tests, and they turned out to be not only not insulin resistant, they're actually insulin sensitive. And if you look at the diabetes, in the Ecuador population, the prevalence is the expected 5%. And in this group, thus far, we haven't seen a single case of diabetes, right? So they're very much protected from diabetes. And we were more confident than we will have been about that when we knew that John Kapczyk has shown something like that for mice, right? So the growth hormone receptor deficient mice are protected when in diabetes models of these mice are protected. So then we ran the clinical trials. They're insulin sensitive. And then what about cancer? We also knew from the work by Kapczyk and Barkey that the mice, growth hormone receptor deficient mice, were protected from cancer. And it turns out that thus far, only one person has died of cancer out of these 100 people that have been followed for the past 30 or 40 years. And also, Zvi Laron in Europe and the Middle East has followed another 250 of these subjects by surveys, and not a single one of them has reported, there is no report from their doctors that any of them died of cancer. So this is really remarkable. Out of 350 people followed for 30 or 40 years, one single cancer death. When, if you look at their relatives, they all living in the same houses, eating the same food, they have a 22% incidence of cancer. So it's definitely not an environmental factor. Okay, so then I didn't talk about proteins, but we just published a couple of years ago a paper showing that, in fact, the proteins are the major regulator of both the growth hormone IGF-1 signaling and the TOR cis-kinase signaling pathways, right? So then we looked at people having high protein diet and low protein diet, and in fact we showed that people that were on a low protein diet were protected at their lower risk for cancer and potentially diabetes, and also lower risk for overall mortality. So this, together with a lot of other data, suggesting that the inhibition of these pathways can lead to reduced aging rate and reduce diseases. I'm not going to talk too much about this because we're still working on that pathway. Okay, so then what about dietary intervention? So a lot of the times, I was always surprised because when you look at science, we're always talking about things that are going to happen 20 years from now, 30 years from now. And then when we first started working with fasting, we were getting emails from cancer patients saying, you know, I have the problem now. What can I do now, right? So I was always very surprised that there's almost nothing for now, right? There's always things for the distant future. And So we had known for decades that if you take yeast and you switch it to water, and this is something, one of the first experiments I did as a graduate student, they live, not only they don't starve to death, but they live longer, right? And they become very resistant. So this always stuck with me. And so I started thinking, well, is there a way to exploit this? Is there a way to control these genes that regulate aging by a dietary intervention? And the first thing that I started thinking was this extreme stress resistance that yeast acquire when they're switched from sugar to water, right? So they become very resistant. And I also started thinking, well, what would be the disease where we can utilize this the most? And I thought about cancer. Chemotherapy is probably the one toxin that we're most likely to be exposed to in our lifetime, right? Or almost 40% of people will be diagnosed with some type of cancer. So then I said, well, let's use it for chemotherapy, right? It's a chemotherapy resistance. And we knew that the fasting caused dramatic changes in glucose. And so remember the glucose pro-aging pathway, and also IGF-1, 70% decrease in IGF-1, and a 10 to 20-fold increase in IGF-BP1. So if you starve a mouse or a person, you have this dramatic reprogramming of the whole system, and it seems to be a reprogramming that is very much consistent with the stress-resistant profile. These growth factors go down, sugar go down, and the system gets into this protective mode. At least that was the hypothesis. And so the first experiment was done in mice, and Litzia and Cheng-Gan Lee, my lab, performed those. And the experiment was very simple. If what we knew from yeast was true, then if we starve mice, we would be able to protect them from chemotherapy, but the cancer cells will not be protected. Why? Because if you remember what I showed you are the proto-oncogenes, so the genes that are then mutated to become oncogenes, they're the ones that control stress resistance. So by definition then, the oncogenes in any cancer will shut down this protective system. So it doesn't matter what you do, if you starve the cells or not, they'll keep being in a sensitized mode, right? So the oncogenes are always on, you cannot shut them off, but the normal cells will respond by being protected. So you starve, normal cells are protected, cancer cells, because oncogenes are on, cannot be protected, right? So this was the hypothesis that we would be able to exploit. And so at the beginning, we always start with yeast and say, well, if this is true, it should work with yeast. And I just summarized the experiment without going into the details, but basically it was the following. You take a normal yeast and you starve it, and then you take the same normal yeast but have a constitutively expressed oncogenes, right? So RAS Val19, so where to mimic a cancer cell. And the idea was, if you then mix the normal yeast with the cancer-like yeast in the same flask, and then you use chemotherapy, you should be able, even though they're mixed together, you should be able to just kill the cancer-like cells, right? The ones expressing this RAS Val19, this constitutively active RAS. And in fact, that's what happened. This is MMS or cyclophosphamide, and you see here are the cells that are mimic fasting, they are also fasted, and these are the cells but these are normal cells. And these are the cells instead expressing the oncogene-like RAS Val19. So now the alkylating agent can kill specifically only the ones that express the oncogene-like RAS, and this is also the case for cyclophosphamide, a widely used chemotherapy drug, right? So, and this we call differential stress resistance, the ability to protect only normal cells that are obeying and not cancer cells which are disobeying. And then we did this in mice, as I mentioned, and it worked extremely well. So this is cisplatin, and these are the mice that were fasted before chemotherapy, and these are the mice that were on a regular diet before chemotherapy. And this is doxorubicin, mice fasted before chemo, mice on the regular diet. So it's really dramatic differences, and I'll just show you a video. I don't know why this jumped. if it works. Yeah, so on the left are the ones that were fasted before chemotherapy, so they're moving around normally. And on the right are the ones that received the normal food, as most patients around the world do, and as most oncologists recommend. So a big difference between the two. And of course, the one on the right, 65% of them will die in the following 10 days, whereas almost none of the mice that were starved before chemo died. Okay, so then as we were approaching the oncologists, they all say, well, so what? What if you're probably also protecting the cancer cells? And we thought, no, we don't think so, but they say, show it to us. And so again, yeast, we turn back to yeast, and the idea, again, is simple. And if you have yeast in glucose, whether they have an oncogene like RAS or not, it doesn't really make any difference. It's heat shock, hydrogen peroxide, menadione. But then if you starve them, and you have cells disobeying by having this oncogene like gene always on, then the starvation together with the stress makes the cells, the cancer-like yeast cells, very sensitive to stress, right? And that's what we call differential stress sensitization. Why? I use the analogy of somebody in the desert. It's very hot, you're in the desert, somebody keeps running without water, you have a problem, right? So maybe if you have plenty of water, you're fine, but you have no water, you gotta stop, go in the shade, right? And the cancer cells, instead, by disobeying, they get in this situation that there's no sugar, or there's very little sugar, but yet you keep having these oncogenes pushing you to grow, and it's just not sustainable, right? And so we thought it's just such a simple concept, maybe it work. And it does work, at least in mice. And so here, just one cycle of chemotherapy plus fasting, you're starting to obtain about 10% of the mice that are cancer-free, right? And in this case, a couple of cycles, you get 40%. This is breast cancer, this is neuroblastoma. You get 40% of the mice that are cancer-free after 6 months, right? So it's really able to, the combination of the stress, the high stress, and the starvation condition really makes it very difficult for any cancer cell to survive in the mouse. And this is a study by a group in Switzerland from a few years ago. And I just focus on these two panels. This is lung carcinoma and mesothelioma. And basically, this is showing the percentage cancer-free survivors, right? So if you do nothing, all the mice die. Cisplatin, all the mice die. Fasting, all the mice die. And then you have fasting plus cisplatin. And now you have 40% of the mice that are cancer-free. And same thing here for mesothelioma. Combination of the starvation and the chemotherapy is now getting about 60% of the mice to be cancer-free. So to summarize this part, you starve a system, you starve normal cells, a mouse, and we believe also humans, and I'll show you some data. The IGF-1 growth factor goes down, IGF-BP1 goes up, glucose goes down, and probably, and we're doing a lot of work on this, probably through these transcription factors that you heard about that are protective, the cells go into a protective mode, you reduce growth and focus on protection. In tumor cells, it doesn't really matter where the oncogenic mutation is. The main thing is they're, by definition, disobedient, and they're just going to keep on growing, and this makes them not only not protected, makes them sensitive. So now you have protection of normal cells and systems and sensitization of the cancer, and that's exactly what you want. Okay, so then this is another experiment done by Chai Wei in my lab a few years ago, and again, as I just showed you, you starve the mice, and this is cyclophosphamide, a drug that hits specifically bone marrow cells and blood cells, and used for a wide variety of cancers. And so again, here you see 6 cycles of the fasting plus chemotherapy. All the mice are alive, and this is the chemotherapy alone, and now you have 60% of the mice dead. But this is not the point. I wanted to sort of start focusing on the bone marrow, and you see here protection of bone marrow cells by fasting, less apoptosis, protection of the stem and progenitor cells by fasting, less apoptosis in them. And then when you look at DNA breaks, if you hit the mice with cyclophosphamide, you have a much higher level of DNA breaks in the cyclophosphamide-treated mice and much lower if it's combined with fasting. But my point is really this. It's not about protection anymore. It's about regeneration. And so as we were doing this, we noticed that after 6 cycles of the fasting plus chemotherapy, the white blood cell number, which was not protected by fasting. So stem cells are protected, but white blood cells are not. And so they go down after chemo and they stay down, but then after 6 cycles, they go back to normal levels, right? So we thought this cannot be protection, it has to be regeneration. And also if you look at the lymphoid myeloid ratio, is nearly going back to the normal level. So we start looking at the possibility that fasting actually can turn on the regenerative system in the mouse. And so this is the hematopoietic stem cell system where you have long-term hematopoietic stem cells, HSCs, that are self-renewing and they can give rise to themselves, basically. And short-term HSCs, progenitor cells, and then they can split into either into the myeloid cells or lymphocytes. So we first looked at, does fasting increase stem and progenitor cells? And it does. But more importantly, we did transplants. So we took bone marrow cells, either from cyclophosphamide-treated, chemotherapy-treated mice, or chemotherapy-treated mice that also were fasted before the chemotherapy, and then transplanted into irradiated mice, and then looked at how competitive, how functional are these bone marrow stem cells. And you see here, after 1 month or 4 months, the stem cells from the fasted mice are much better at generating peripheral blood cells. So they're not only higher level, but they're also functional and able to give rise to a functional immune system. Okay, then just very briefly, we've done a lot of work on the mechanism, but certainly growth hormone deficiency, again, not only is involved in protection, but it seems to be also pushing the system to regenerate. So when we use growth hormone, these long-lived growth hormone receptor deficient mice, we saw the same trend where after so many cycles of chemotherapy, the white blood cell number goes back to nearly normal because of this IGF-1 and probably insulin deficiency. Okay, so fasting reduces growth hormone receptor IGF-1 signaling and increases hematopoietic stem and progenitor cells, which leads to white blood cell recovery. And just, of course, we did a lot of work on mechanism, but I'll just show you the key take-home message, which was by inhibiting either IGF-1 or inhibiting PKA, and remember PKA from the yeast work that is very central in accelerating aging, but it seems like in mammalian system, the inhibition of either IGF-1 or PKA is causing a long-term effects on the stem cells, increasing the stem and progenitor cells that have long-term effects on the peripheral blood. So that means that the same genes that are accelerating aging are also blocking regeneration of the blood system. Okay, so then summary, just added this PKA, and we think this FOXO1 and G9A genes are implicated in these effects of hematopoietic stem cells, in the activation of hematopoietic stem cells and the regeneration of the immune system. Okay, so then very briefly, just a few months ago, then we published on if you regenerate and rejuvenate the immune system, is it possible? Now we know about immunotherapy and how effective it is in cancer treatment. So we started thinking, well, is it possible that it is the starvation, as I just showed you, turns on the immune system, and it is the immune system to attack the cancer cells? And we started with a cancer, breast cancer, which is not particularly sensitive to this immunotherapy. It's not being one of the cancers for which immunotherapy has been successful. And first of all, we actually developed what we call a fasting mimicking diet. So we realized that patients do not like to fast, and oncologists do not like their patients to fast. So we come up with a fasting mimicking diet that it's calorie restricted, but it's got about half of the normal calorie levels. And so you see here, if you do 3 cycles of the fasting mimicking diet with doxorubicin, with chemotherapy, this is the breast cancer progression. These mice do the best. And this is the mice, the control mice. But the most important thing was if you look at tumor-infiltrating lymphocytes, only the combination of the fasting and the chemotherapy causes a major increase in the T cells, the cytotoxic T cells now being associated with the tumor. And also a decrease in the level of regulatory cells, that are inhibitory cells at the tumor site. And then when we blocked the T cell production by using this antibody, you see that this is sufficient to cause the tumor to be much more aggressive and grow much faster in the presence of the FMD. So this is suggesting that the cycles of this fasting mimicking diet are delaying tumor progression in large part by activating the cytotoxic T cells. You remove the T cells and the effect is not there, or is much, much reduced. So, then I'm just going to skip this slide. This is about mechanism. We figured out that this heme oxygenase 1 that breaks down heme, this enzyme that breaks down heme, is lower in the fasting, and this lowering is key in these effects on the immune system. And I'll just summarize it here, which is normally you have very few tumor-infiltrated lymphocytes and these breast cancer cells, and you'll have a lot of the regulatory cells. And you have a high level of heme oxygenase 1, and during the fasting, these Tregs are replaced with tumor-infiltrating lymphocytes thanks to this lowering of heme oxygenase 1, and now the immune cells can kill much more effectively the cancer cells, and we're now trying to figure out much more about the mechanisms responsible for this. Okay, then what about humans? And we started a long time ago, about 10 years ago, we started going to oncology, the clinics, and trying to figure out, because a lot of patients were contacting us and saying, I'm fasting in combination with chemotherapy. And so we started collecting the data. It actually turns out in a case report, very preliminary, years ago, that patients fasting in combination with chemo, we're reporting less side effects, whether it was fatigue, weakness, headaches, nausea, vomiting, etc., compared to those that were eating a normal diet. And then we just finished an 18 patient, again, preliminary trial where we looked at 72 hours of fasting compared to 24 hours of fasting. And the 72-hour fasting is associated with reduced side effects in all kinds of different criteria. And Leiden, University of Leiden, also did a small trial. This was randomized with I think about 13 patients, and showing effects of the fasting and protection of erythrocytes and thrombocytes. So that's also promising. And Charité Hospital in Germany, in Berlin, finished their trial with 34 women with breast and ovarian cancer. And this was a randomized crossover trial, and the results are very positive, and they haven't published it yet, so I cannot show you the data. But so this is encouraging. Okay, so then going back to aging, so this is working very well with stress resistance. You starve the mice, so you give them this fasting mimicking diet, and they're protected. And of course, we were interested. Well, can we use this to extend healthy lifespan? And so we did this. We took mice that were middle age, and again, we were really in the aging field. Many, including us, are working on drugs, right? What are the drugs, like blocking the growth hormone receptor? We're working on that, but it's going to take probably 20 years to get there. And so we started thinking, well, what is something that could be done now? And certainly this fasting mimicking diet, we thought it was something that could be applied almost immediately if it was clinically tested and shown to be successful. So we started with the mice, and so middle-aged mice given this diet twice a month for 4 days, right? And that was also very important to us, to come up with something that people could actually do, right? That wouldn't necessarily impose a complete change in someone's diet. I mean, most people, we spend a lot of time trying to change people's diet, and most people don't want to change their diet at all. And so in the 4 days, in this case for the mice, I'll show you the human data in a second. And the first thing was interesting that the mice, even though they were on this low-calorie diet for 4 days, twice a month, per month they ate the normal amount of calories. So they were not really overall calorie restricted, and yet they lost a lot of weight. And you may have heard there was an article in the New York Times recently talking about these extreme diets that actually slow down metabolism. But it turns out that this periodic fasting mimicking diet done in the way that I described, at least for the mice, not only they don't slow down metabolism, most likely they either accelerate metabolism or promoting this reprogramming. And we know that because during starvation, after a few days, the system starts burning visceral fat. As you can see here, when you look at the visceral fat is where we see the biggest difference. So these mice, and this makes sense, right? During starvation, there is no sugar coming in, and the system is starting to take the energy from the depots, and the visceral fat is the largest one, right? So this switch to a visceral fat-dependent metabolism is probably key in these weight loss, these long-term effects on weight loss that I just showed you. And then we did a lot of things, but I'll just tell you a few because I don't have time to go through all of them. But the immune system that I just told you about, there is in mice, between a 4-month-old mice and a 16, 20-month-old mice, there is a major decrease in the number of white blood cells. And you see here, after just 4 months on these cycles of the fasting mimicking diet, the level of white blood cells goes back in a 20-month-old mouse, goes back to the level of a 4-month-old mouse, suggesting that the system is getting rejuvenated, and this is also supported by the lymphoid-myeloid ratio, not going back to normal, to the youthful level, but certainly being much better in the fasted mice than in the mice on the control diet. And so, of course, there is, as I just showed you, a tight link between immune system and cancer progression. So we looked at cancer, the tumors, and you see here the mice on the FMD cycles, they have nearly half of the tumors compared to the mice on the regular diet. And if you look at when they get the tumors, everything is postponed. So you see here the gray dots are the tumors being detected in the mice on the normal diet, and the red dots are the tumor in the mice on the fasting mimicking diet. You see that everything is postponed. And if you also look at the type of tumors, the mice on the FMD have most of the lesion in 2 or less organs, and the mice on the control diet, a lot of them are 3 and more organs, suggesting that the FMD reduces the tumors, postpones the tumors, but also switches them from malignant to benign. Right? So it's really powerful effect of this periodic diet. Then the effect on health span is the biggest, actually, at the 75% point, and then it becomes 11% lifespan extension as a mean lifespan, and then it disappears by the maximum lifespan. And we suspect, as we've shown for protein intake, that there are different phases of life, and that mice, and we actually know that mice that are very old don't like to be put on these extreme diets. So the same thing that works up to a certain age in a very old mouse is probably detrimental. So now we're trying to understand how we can keep the benefits that we see up to in all mice, and maybe have this also affect the maximum lifespan. Okay, then of course we develop a fasting mimicking diet for people, and this again is about 50% of the normal calories, and it's high fat. It's a high fat diet, low sugar, low carbohydrate diet, and so we ask people, 100 people. We enrolled them, and 71 of them went on 3 cycles of this diet. It's 5 days long, once a month for 3 months. So we measure baseline, and then we measure everything again 1 week after the 3rd cycle. So we wanted to do most of the measurements when they're on a normal diet and not when they're on this fasting mimicking diet. It's a vegan diet, mostly nuts and vegetables is what's composing this diet. So high fat, good fats, and high vegetable. And I just show you a few slides on this, and then I'm done. And we showed that this is from the 38 patients. Now we're up to 100 patients, but this is the results from the 38 patients. And you see, as I showed you for mice, trunk fat is specifically reduced, but no effect on lean body mass. In fact, the relative lean body mass goes up. And we believe that this is due to the regenerative effects of the diet that we've shown actually in mice and the muscle of mice. And if you look at IGF-1, one of the markers and potential risk factor for cancer associated with many different cancers, levels of above 200 are associated with many different cancers. There is a major long-term reduction in the level of IGF-1. C-reactive protein, a major inflammatory marker associated with the risk factor for cardiovascular diseases. And almost every patient that had a high risk or moderate risk level of this C-reactive protein moved back to the either normal level or nearly normal level. And we believe that, as we demonstrated for mice, that this the increase in the regenerative in stem cells and the regeneration in multiple systems is largely, this is speculation. We don't know that. We know it from mice, but we have no idea for people. But certainly this slide makes us think that this is much more about regeneration and rejuvenation than blocking anything. So for example, if you look at BMI, the effect of the 3 cycles of the FMD on people that had normal weight is not very high, but it's much higher on the ones that were obese to start with. Effects on systolic blood pressure, not big on people that have normal blood pressure, but much bigger on people that have that are hypertense. Same thing for diastolic. IGF-1, relatively small effect on people that have low IGF-1, much bigger on people that have over 225 IGF-1 level. Glucose, no effect on people that have normal fasting glucose, but an effect on people that are pre-diabetic. Same thing for triglyceride, cholesterol, LDL, C-reactive protein, right? So, I mean, there's many explanations for this, but we think the most likely is that by fixing a lot of the organs, or certainly helping to fix a lot of the systems, everything is being normalized. You know, so if you have a low level of blood pressure and the system is already working very well, and this FMD refeeding cycles probably don't do very much. And why does it work? And I use my a picture of my PhD mentor, Roy Walford, who was one of the pioneers of calorie restriction, to sort of explain this in a very simple way, which is Roy, this is Roy before entered something called Biosphere 2. So there's 8 people, including himself. He was a UCLA professor who in 1990, I believe, had a bad idea to lock himself up in a place called Biosphere 2, and they became calorie restricted in this Biosphere 2, right? So they ate about 1,800 calories a day, and as you can see, this is very extreme. And but the point I'm trying to make, that if you look at Walford before he entered and Walford here, most of his organs, for example, his liver here was probably about half of the size that it was before he entered Biosphere. So now you have a liver that goes down to half of the size. But not just the liver, the muscle, the heart, everything now is much smaller. And we know this from mouse studies, but obviously you can tell also by just looking at this picture. Then he exits Biosphere 2, he feeds, and his organs now return to normal size, right? So we believe that this is probably the most powerful intervention available to reactivate these programs that are normally activated during development. And I showed you one example with the hematopoietic system, right? So the white blood cells during fasting go down, the stem cells are turned on, and then when you refeed, that's most of the effects is not happening when you fast, it's happening when you refeed. Now during refeeding, everything is now being regenerated, whether it's the muscle, the liver, the blood system, and even the brain. We're showing cells in the brain, stem cells in the brain being activated. So we think this is probably one of the most powerful ways to reactivate this natural rejuvenating system that is able to turn on the good stem cells and rebuild the system. I mean, of course, there are limitations. It's not like becoming a brand new system, but certainly there's a lot of evidence that I just showed you that, and the patients in the trial were 20 to 70 years old. So a lot of evidence in all age ranges that you can rejuvenate at least a little bit the system, and that rejuvenation can have major effects on risk factors for diseases. And I already thanked the people, so that's all I have. Thank you very much.
Thank you very much, Walter. And since we are running out of time, I suggest we have a very quick coffee break and peeing break, and then we come back in time and write down your questions and keep them in mind for the discussion. Thank you.
Okay. Welcome back. So we continue now, and the next talk will be given by Professor Elin Slagboom, Head of Molecular Epidemiology, Leiden University Medical Center, the Netherlands. Professor Slagboom obtained her Master of Science in Biology in 1987 and her PhD in Biochemistry in 1993. Between 1993 and 2000, Slagboom was investigator of Department of Vascular and Connective Diseases Research at the Glaubius University in Holland. She initiated a unit for genetic epidemiological studies aimed at the identification of genetic determinants and multifactorial diseases. She was in 2000 awarded a big program, a big grant from the Netherlands Research Council, and was in the same year appointed professor in molecular epidemiological disease at the Leiden University Medical Center. She established there a new section of molecular epidemiology and a genotyping center. Focus of that research is to use omics data, genetic, transcriptomic, epigenetic, metabolomic data in molecular epidemiological studies to find biomarkers of aging and age-related diseases in human cohorts and to understand the pathways underlying these complex traits. A diversity of human cohorts with unique study designs are being analyzed for this purpose. Slagboom is also co-founder of the Research of Aging unit in Leiden, and she initiated and is PI of the Leiden longevity study, where various research cohorts of patients are being investigated. She serves on many national review boards, including the Netherlands Research Council and various other organizations, such as Dutch Heart Foundation, Scientific Advisory Board, and she is also a member of the editorial board of Aging Cell. So with that, I would like to welcome you, and we are eagerly looking forward to your talk. Thank you.
Thank you very much for that kind introduction. It's funny because whenever I say I'm professor of molecular epidemiology, then my husband always says, "Do you want coffee?" It's really a difficult word, epidemiology. So thank you for the organizers to give me the opportunity to talk about human research in aging and longevity as we do it in Leiden and in the Netherlands. And I want to linger on a couple of topics today, and they are mainly from the perspective of the elderly person. So it's not about healthy aging, how to avoid metabolic syndrome when you're 40. That's, I think, another ballgame. It's healthy aging, what can you do in the second half of life? So on nature and nurture of healthy aging elderly. And the things I would like to discuss is to discuss what is actually healthy aging in humans. And we have studied individuals from families where many people become very old. And from them you learn what healthy aging actually is and what kind of benefits do you get. And then the conclusions that we draw are of an impact for the hospital, because in the Leiden University we are in a unit with about 20 professors where all of us together want to improve the health service for elderly individuals. And of course, any kind of study will start by trying to find causal factors for healthy aging, maybe in families or in large populations. And that is looking for determinants by genetic and genomic studies. But of course, that would be nature. Nurture is when environment hits in. Environment hits in early in life, late in life, having an impact on healthy aging. And if you want to follow those things in the clinic, and if you want to understand whether somebody that you're looking at is a healthy ager or not, you want to have biomarkers. And then in the end, you also want to use those biomarkers to intervene in the healthy aging of individuals and to monitor whether they're doing well or not. So basically, the interesting thing, especially about aging is, of course, is that age is the major risk factor for almost all common diseases. Right? So something happens here in this period of life that is influencing the risk for all of these diseases. And it's fantastic, of course, that in the last 200 years we have immensely increased in our lifespan. And so it's even ongoing. So at 2050 in Europe, the population of people above 65 is expected to double and of 85 to triple. And the clinicians actually, they don't almost even dare to look at that because it's making life very complicated in the clinic. Because although we're living so long, 20%, 15 to 20% of our lifespan will be spent with disability. So you will end up with difficulties. And so thinking about aging and the aging population is really thinking about the comorbidity. What factors are happening somewhere here in midlife that are contributing to your risk for different types of diseases? And could there be switches? Could there be things that you change in this period of life, or maybe even earlier, that would change the situation? The use of medication for so many different diseases, the heterogeneity, right? So people with a heart attack when they're, let's say, 45 years of age, may be a bit more homogeneous in their causes. We can find them, we can find the mutations, but all the people that have these heart problems at 70 or 80, they have these problems when they have also many different problems. So it's a sick heart in the presence of a changed body. And how can you treat that? So the heterogeneity between individuals is large at older age. So what is the biological age of individuals? If you put 10 80-year-olds on a row, are you going to treat them all the same? If you know that some of those 80-year-olds are physiologically as if they were 95 and others are actually as if they were 60, are you going to treat all these people the same? So these are typically questions that you talk about when you're focusing on the elderly population with clinicians and biologists and many different medical professions. So if this is the survival curve, then you have a period where people are the first to die, and then a period where many people die, which in our case would be between 60 and 80. Not all the people really become that old. And then there is this tail of people that become very long-lived. And now from a genetic point of view, it's logical to pick out these type of people and these type of people to study. Because within their families you expect that genetic factors can be found, can be isolated, and can be investigated. Whereas, of course, you always want to say a bit more about the population as a whole, and that is exactly the most difficult part in all our research. So, in our case, we did not focus on the metabolic syndrome, we focused on longevity families. Here you see 4 sisters, and they came in our study when they were all above 90 years, and 2 of them are now actually above 105. And here you see my favorite couple. This is a monozygotic twin, and they're 103 years of age. And whatever you look into their metabolic health, their lipid metabolism, they're extremely concordant still after 103 years of life. One died now, unfortunately, but if you look into their families, there's definitely a high heritability of longevity. So whereas for all of us, as a population, the heritability of our lifespan is almost not more than 30%. So the environment has a major impact on the lifespan of all of you. But if you go to these families, now the importance of the genes really increase. Many more people become very long-lived. And so we decided in about 2000 to start collecting these families. So we collected people above 90 years of age, with brothers or sisters that were also above that age, and we collected in the end about 1,000 of them. Yeah, and that is the way you want to collect the people, but you cannot do so much research with them. You know, you cannot put a 100-year-old for fun in an MRI. right? But you can do it to their children. So, but the children in this case, so the children in this case are people of about 60 years, right? And this is called the offspring generation. So you wonder, how is the aging phenotype of this offspring generation as compared to their own spouses with whom they share lifestyle? They usually share already 40 years of eating together and living together. But the difference is that these people are offspring from long-lived parents, and so you hope they have the long-lived phenotype, and these can serve as controls. So this is the study design that I will talk about. This is called the Leiden Longevity Study, and in total we are investigating 3,500 people here. So what is the phenotype of healthy aging? Well, basically, if you compare the offspring to the spouses, and in the Netherlands we call the spouses the cold side, and so, you know, to the despair of the cold side, the offspring from the long-lived, they have less myocardial infarction, less hypertension, less diabetes, they use less cardiovascular medication, a lower prevalence of metabolic syndrome, a lower prevalence of osteoarthritis. Somebody mentioned it yesterday. I thought it was fantastic to find this, because osteoarthritis has always been such a wear and tear type of disease, you know, what to do about it. It's something so much linked to the aging process itself. And my lab does a lot of work in this field. This is the joint disease. But actually there's such a metabolic component in osteoarthritis, so that it's really got a lower prevalence in these families. So that's the kind of bonus that you get from being born in these families. And at the point that this table was made, the malignancies were not different. We have now data just collected and finished from 15 years of follow-up of these people. So we are now going to investigate whether also the malignancies are present at the lower prevalence. And then it turned out that if you compare the offspring to the spouses, you know, we expected that they might be healthier or more lean, less fat, they might eat different. It's not the case. So you can test by a food frequency questionnaire whether the people from, so the offspring from these kind of ladies as compared to their own spouses, if they're different, they're not. Physical exercise, they're not. BMI is not significantly different between offspring and spouse. But if you look at a more intrinsic level into their metabolic health, then you see differences. They're more insulin sensitive. They have lower serum insulin, lower triglycerides, larger LDL particle size, lower blood pressure, high adiponectin, lower serum free T3, which is the thyroid hormone. Actually, all of these things we would call things about metabolic health, because people with metabolic syndrome, they're actually on the other side of the spectrum. They're doing exactly the opposite as these people are doing in these families. And then other types of bonuses are that even if you live with a cytomegalovirus infected partner, they're still resistant to this kind of infection. So their immune system is different. If you take the cells out of their bodies and you expose them to damaging agents, they're more resistant to damaging agents. And so the picture that comes out of this is that if by nature you have this intrinsic metabolic health, wow, look for what kind of, let's say, problems you would be protected against, right? So metabolic health, to all the doctors in the hospital and also the biologists, is really a major thing to start collecting data on, because it will give an insight into the consequences for many different diseases. So when I talked at the beginning that I'm so interested into common denominators of different diseases, well, metabolic health and the things that regulate all these factors, those would be of major interest to me. And I can tell you up front that if you think about it, you would have to look into the muscle, because what's going on in the muscle is of major impact of the metabolism in your whole body. You have to look into the brain, in the thyroid metabolism, equally involved in the metabolism of the whole body, but also the gut microbiome and the fat, right? So from this point of view, you realize that it is a shame that in the hospital they're not collecting all this stuff like they're doing, let's say, in an epidemiological study. Yes, keep that in mind, that it had an impact when we had these findings. And that is why in the clinic we then said, how can we integrate what we're doing and, let's say, put metabolic health or immune metabolic health in the middle, and put also in the middle that we want to know about everybody above 50 years of age, what is your biological age? Not what is your chronological age, but what is your biological age? And if I knew, would it change my decision how to treat? And so what needs to be integrated are these things. So this is just quickly to get a bit of the perspective of the physicians next to the biologist. They would consider that elderly people that come into the hospital, it is always about neurocardiovascular problems and about musculoskeletal. You know, so if somebody comes in with a heart attack and that person is above 70 or above 80, it would be good to do some brain testing, because there may be a problem in the vasculature, and after treating them for the heart, they will be back in a year for the brain. Yeah, so that's become one kind of area in aging research of elderly people. The other side is also true if you start treating them, but they can't move because they have sarcopenia, muscle weakness, or they have a lot of joint problems. You can treat them, they go home, they don't move, they're back in the hospital. Yeah, so if you just focus on any of these alone in the elderly, it's really a suboptimal treatment. And so to work together, you really have to now integrate different types of research. And so I'm going to start by talking about how to understand metabolic health. What did we try to do about genetics? And during my talk, I will also linger on how can we go beyond blood, because I'm talking here about healthy people. And of course you can do imaging and a lot of stuff, but if you want to look into the tissues, and normally we have only blood, but we want actually also other tissues. In the clinic, they're dying to make classification, like I explained. Classification, maybe not so far as personalized medicine, but at least to be able to stratify people. That if you're going to give people an operation, let's say a hip replacement, of the elderly that get a hip replacement, about 25% is not happy, is not working, they get sarcopenia, they get a lot of problems. So would it make a difference if, let's say, Lee had his way and he could help us in making 5 strata of people the moment all the people come into the hospital, and you would say, you know, strata numero 4 and 5 is never a problem. Treat them, they won't get sarcopenia. But 1 and 2 is a problem. So there you have to put all the effort after the operation to make sure that they move and to coach it, because otherwise they will develop sarcopenia. Elderly people can lose their muscle within a week, you know, and you can't regenerate it, because all the systems we were talking about, they can't be excited so easily in the muscle of an elderly person. So you get in a lot of trouble that you may have prevented if you knew that this person is at risk. And then, of course, if you do the interventions, so if you think about, we talked about the intervention that Walter is doing, if you do that kind of intervention on an elderly individual, on a 70-year-old, I guarantee you that a number of people is not going to respond very well. Also, medication is not, so all medication is really tested in younger people. So if you apply them to 70 and 80-year-olds, a number of people is not responding well. And so with all the interventions, you also want to use everything you learn about metabolic health to monitor what's going on, right? So it's always this triangle what people are talking about. So first linger a bit about the genetics. I want to find the genes that make these families live longer. And we apply GWAS studies, so all of these red dots are markers, genetic markers, all across the chromosomes, and in a very systematic way, you're testing where can I find an allele frequency that is different between these kind of people and controls. So you do a lot of elderly and you compare them to controls. Well, if I do that in the light longevity study, it's too small. And so by doing the genome-wide association scans, we all learned it has to be bigger, and actually that's what we did. So in the end, we did a study in about 20,000 people of ages above 85 years in all of Europe, many different studies. And it was all done by Joris Delen. He kind of supervised it. And what he's doing now is a worldwide effort like that, with different types of consortia in China and in the US, the Church Consortium. And when he did this study, so he did a GWAS study for longevity, trying to find out at which locations in the genome can I find genetic variants of which one of the alleles is significantly increased in the elderly or decreased. And then, well, you already, you will always find ApoE, you know that now. FOXO3A is difficult because you usually do not find it in genome scans. The Church Consortium has found it, we didn't find it, but it's definitely an important gene. But we did find a novel location. We found variants at chromosome 5q33. It's difficult to talk about it because it's a location. It's not in the middle of a gene or something. And carriers of that minor allele, they have decreased mortality risk from middle age onwards. So really, if you follow, this is actually a study where it was followed above 85 years for 10 years, and you can see that carriers with this allele, they live longer. That's a prospective study. But we also saw in other populations that if you start following mortality from middle age, you see that the carriers always live longer. They also have decreased blood pressure from midlife onwards. But that doesn't explain the mortality effect, so there must be more. Carriers actually show interaction between this blood pressure and BMI. Now, of course, there's for everybody an interaction between blood pressure and BMI, but we think that these people may be more protected, that if they have higher BMI, they have still rather low blood pressure. Then the SNP associates to serum LDL, but also to the response to statins. And it also, and because for many different diseases, people are doing these genome-wide scans, and you can look them up in a catalogus. So we looked up our SNP, our genetic variant, and then it turns out that it's also associated to neuroticism. And that actually links to what is happening there. We have a genetic variant, and very close to it is the early B-cell factor 1 gene. When we first heard it, we thought, early B-cell, I don't know, maybe immune responses or what. But now other people are studying now this gene, and more information comes out. Now we have a new starting point to study this gene. There are also other genes in the neighborhood of this genetic variant, but this one becomes very interesting at the moment. So you're now looking into the environment, whether there are enhancers in the neighborhood, and it's becoming known now that the EBF1 is involved in adipogenesis and in early development. It's associating with let's say, metabolic and cardiovascular phenotypes, and it's all the time dependent on stress levels. Stress levels, BMI, right? So this is an interesting gene because it may give you the advantage that you need to become very old, especially under stress levels. Stress levels, high BMI, so in interaction with environmental factors. So what we're doing at the moment in very large studies is to look into this gene, into the epigenetics, etc. What else can you do from a genetic point of view? You can, of course, sequence. So we have sequenced our favorite ladies, but also 220 of these people from the longest living families. And if you do that, you get no answer, because every individual has so many interesting functional variants that by this number of people you cannot figure out which kind of variation is associated to their longevity. So we're teaming up at the moment with people that are doing the same, for example, in Utah and also in other studies. And there's one thing that may help us is that we've entered historical data from the Netherlands from 1750 onwards. So we're now looking at people that were then already long-lived with their family trees that now are still long-lived. So you have much better family information to help you into the genetics. And we did a lot of transcriptome and physiological studies, and I don't have time to go into that because the major point I want to be making is that for the Leiden longevity study, everything pointed all the time to energy metabolism. nutrient sensing. You've heard about it. The insulin signaling is different, the TOR signaling is different, and all we know about looks into the direction that it is more healthy, that it is responding better to the environmental stimuli. So for the longevity study and the metabolic health, we are always very focused on lipids, insulin signaling, thyroid signaling, and the TOR pathway. So if this is my story so far on the determinants, we have to realize that in the population at large, only 30% of lifespan is heritable. So what can we say about nurture, about the environment? Well, one thing is for sure, this is the island Okinawa, where there is the highest condensed region of long-lived people, of centenarians. And of course you can say, well, that's a genetic isolate, so they are genetically like each other and they're more healthy, maybe. But also the other factors come together there. You know, they eat healthy, they live healthy, they're not lonely, because they have a close community also when they're older. So many, to become a centenarian, many other factors than just your genotype matters. And if you go from there, it's good to know that there has been, for example, there have been multiple studies, but I want to mention one. 20,000 individuals were followed for 14 years, and the difference in lifespan or life expectancy between the people that scored the best on healthy behavior and the worst is 14 years of lifespan. Yeah, and this was just talking about smoking, intake of fruit and vegetables, intake of alcohol, and physical activity. So a part of the 70% environmental influence on longevity is definitely your own behavior. The question is, of course, you need to behave well from early life onwards, but the question is also, when you become a little older, I'm 56, I'm already starting to understand that life has an end. So you may want to do something about yourself, and definitely people between 60 and 70. They can be highly motivated, and you can approach them in a different way than a 40-year-old running up to metabolic syndrome. These are not the same kind of people. And so the question is all the time, can you improve your metabolic health? To linger on nurture, there's also a very strong indication that early life matters, whether your mother was an overweight mother. And in our case, we have investigated people that were in the uterus during the Dutch hunger winter, when the mothers underwent serious shortage of calorie intake. So the children that were then in the uterus, they were 60 years of age when we investigated them. And as you can see here, that the individuals that were almost conceived in the Dutch hunger winter, they suffer 60 years later from schizophrenia, higher rates of extreme overweight, and some of the problems. And mothers that were already pregnant when the hunger winter hit in, those children are overweight, diabetic, etc. They suffer less from schizophrenia. So these are late effects of early developmental problems that humans are sometimes exposed to. And so, of course, our question was, can you see in these 60-year-olds that there are, for example, differences in the response of the genome to the environment? Yeah, I'm just trying to touch, the genetics is one part of the genome, and then the transcriptome. And in this case, we're talking about how does the genome regulate gene expression in response to the environment. And I can tell you, you can do genome-wide studies, but what you actually, of course you do that, we all do that, but things can be regulated very specifically for specific regions. So you have to bring cells and tissues and humans in different situations to start understanding how the genome actually responds to these environmental situations. And one way, there are many different things in which the genome regulates gene expression. They're very elaborative to investigate. One of the most easy ways is to measure DNA methylation. And in my lab, we are always doing research with, of course, thousands of individuals. So we would always choose to investigate DNA methylation first. And so when we investigated in the Dutch hunger winter individuals, what happened to their DNA methylation, we saw indeed that especially individuals that were conceived during the hunger winter, they had major changes in their DNA methylation that correlated to their decreased metabolic health. Yeah, so also the moment in time where when you are in the uterus, the system can still respond to a hunger situation outside, that time frame is not the full-blown pregnancy. Most of that regulation happens actually very early in pregnancy. And to stick a little to epigenetics, because we're talking here about, molecular markers by which you can follow what happens to individuals during their life, then another part is that if you measure the epigenetic profile of the genome, you can see at all different types of spots in the genome that things change linearly with age. And you can even make an algorithm out of it, so that you can tell exactly, if you would have one cell of an individual, you could tell exactly what age that person is. So there was quite a hype around this, epigenetic clocks being associated to chronological age. It has very interesting aspects, but it's not enough for the clinic, of course, because we want biological age. I do not want a marker to tell me that I'm 56. I want a marker that tells me that I'm so healthy that I'm like a 40-year-old. That's what I would like. Yes, so from here to that kind of marker in epigenetics is something that these guys are studying in my lab. Bas Heijmans, he also did the Dutch hunger winter studies, and Roderick Slieker. And what they have done is, out of all this epigenetics in the whole genome, they're trying to pick those loci that change the most during your life, the increase variation. And then they try to figure out, is it so that the change in the epigenetic profile during life, that contributes to a disease? Is it so that you can use it as a monitoring marker or as a biomarker? And just one point that is made here, because if you take all the loci out of the epigenetic clock that I was just showing you, those loci are not really that much associated to gene expression. And if you take, also people made a clock out of gene expression, those are not associated to DNA methylation. So you're doing something funny to predict chronological age, but it's not telling you enough about what's happening functionally to the genome. The loci that these guys came up with, that let's say show increased variation during life, those loci are interesting because they're losing control across life. They're polycomb regulated loci in the genome. And my osteoarthritis group has then investigated, in specific tissues, whether the loss of methylation at specific loci now starts contributing to the disease risk, and it does. And so you need to get this overview of the genome first, but then you also have to be very specific, and then you also have to have the right tissues in humans and the right knowledge about the patients before you can say, what is happening to what gene and how does that contribute to a disease. And that's why I'm very much in favor of all these different data sets that people are gathering, because it gives you an indication all the time of where to look. Okay. So if that was talking a bit about understanding and a bit up my way to classification, can you use epigenetics, then I want to mention something that would be most logical to use as a biomarker, because we have already risk markers. If I want to predict your risk for myocardial infarction in the next 10 years, then I will use these variables. And then I will be pretty good in predicting that. We call it the Framingham risk score, and it's a kind of a gold standard. If you apply this on a 70-year-old, it says nothing. Again, above a certain age, in the second half of life, things become rather complicated. And now the doctor, he needs to know, so you have high blood pressure. I want to treat you. If you are an 80-year-old, but you look like a 60-year-old biologically, he will treat you like a 60-year-old. But if you are an 80-year-old and physiologically you are like a 90-year-old, he will try to to keep your blood pressure increased, because otherwise it doesn't get to the brain, and you will stimulate dementia in effect. Right? So you need to know what is the cardiovascular disease risk, what's the neurological disease risk if I give a certain medication to a 70 or 80-year-olds. And that's the fastest growing group of elderly in the whole population. They need to know. So that means that we need more of these metabolites. And happily enough, there are different platforms of metabolomics where you can investigate now also amino acids, not only lipids, amino acids, glycolysis-related metabolites, ketone bodies, fatty acids. And so it becomes very interesting to say, every new platform that it has to have a certain quality. So a new platform is fantastic, but it has to be affordable, cheap. You have to, be able to do standardized measurements with it. And you have to do it in many, many populations before you know what it predicts for an individual. So with many Dutch studies, 25,000 subjects, we all used this kind of metabolomics as a tryout, as a proof of principle, for seven different age-related diseases. And so it's depression, dementia, osteoarthritis, cardiovascular disease, diabetes, and we all use the same platform to see if they can be used, if there are any biomarkers. And then it turned out, what we expected more or less, is that yes, there are very interesting biomarkers, but they're also very generic, these ones. So if you are like 70 years of age and you have an increased level, I couldn't tell whether you have more osteoarthritis in a couple of joints or more vascular problems, an increased risk for myocardial infarction. And so for these specific diseases, this kind of generic marker is not so usable. You have to be maybe more specific and get the proteins out of the blood. But to get an estimate of metabolic health of an elderly that has these different diseases under the skin anyway, these kind of markers may be usable. So this is what we're testing. We're testing these more markers of generic markers of metabolic health. We're testing them now in the clinic. And one of the things that I want to show you is, so we have a study with a lot of different cohorts, and in total it's just now about this number. It's about cohorts where people were followed up, and in total 5,000 individuals died during this period in the different cohorts. Cohorts are of different ages. Now we can see that actually everything above this line is a metabolite that is indicative of the mortality risk of these individuals. Yeah, so now you can look at the mortality risk, and the doctor can have a conversation with the people about their mortality risk, and with the clinicians about, should we treat this person? Is this person at higher risk for complications? So that is its purpose. It's excuse me. Well, thank you very much. No, it's hello? It's stopping. And here I see still my... Hey, thank you. Okay, so one example is, for example, here, if you look into, these are all the cohorts with different age strata. And so a cohort here in the green is below 60, and the next is 60 to 70. And so you can see that triglycerides, higher level triglycerides, increases the risk for cardiovascular disease. Yes, it does. But by the time you're between 80 and 90, and by the time you're between 90 and 100, actually a higher level is decreasing the risk for heart disease. The same for blood pressure, right? So it's almost as if elderly people, they undergo metabolic shifts, and now the same kind of markers, instead of a risk factor, become a protective factor. And this has been long observed. It's been checked whether it's just selection, but it's real. So there is something that we need to figure out about the metabolic shifts of elderly people. Okay, so that is the part about biomarkers, metabolic health, metabolic markers. So what kind of interventions are we thinking about and are we doing? So following studies into calorie restriction and that Walter Longo was talking about, we always wondered, can you do the same thing to elderly people? So there had been a trial already, the calorie study. We took one of their conditions and we applied during 13 weeks intervention with 164 subjects that had a 12.5% reduced dietary intake and a 12.5% increased physical activity. And we collected blood at the beginning and at the end, but also muscle and fat. And we applied different challenge tests. We took DEXA scans. We have wearables so that we know how much they moved. And this is a kind of a pilot study to figure out, what things really work in elderly people and how should we measure these things. And one of the first things we came across was that, yes, there are a lot of wearables, but the algorithms tell you not much about the behavior of elderly. So out of the data and all the peaking patterns, you want to extract how much did they move, how much energy did they burn. And the answers were not clear enough, so we hired a lot of high school students who had nothing to do at the moment. And so they had been standing with a stopwatch where the elderly people were cleaning, and it was really almost shameful to look at it. And with a respirometer, so that you could exactly see what are they doing, how is it recorded, and how much are they burning. Right? And that is what we are now using for interpreting our data. Oh yeah, here you see, of course, our favorite sport for elderly people is sitting on the bike. Okay, so what about this intervention? The mean age was 63, 43% were women, and they had a BMI of 27, which is a bit highish, and a normal blood pressure for this kind of age and BMI. So I'm not going to linger here. I'm going to show you, so in 13 weeks, there indeed was a weight change of about 3.5 kilo on average, and that was comparable to that same study in 50-year-olds. So that was the same. The heterogeneity is rather large, as you can see. And we, of course, now recorded what people did, but sometimes there were people that were really exercising and really adhering to the study, and they would lose 2 kilograms, and other people do exactly the same, and they would lose 8 kilograms. So already at this age group, you have quite a heterogeneity. And as I explained to you, when you're 50, losing weight, lowering blood pressure, lowering BMI, everything is good for you. But by the time you're 75, there are already people for which these rigorous changes may not be so good for them. So you want to record how their metabolic health changed. Another change here, blood pressure went down, and especially here in these individuals that had high blood pressure, the same as what Walter was showing. But for insulin, we were quite different from the 50-year-olds. So insulin hardly changed, and it was also, let's say, more heterogeneous than the other measurements. So some people did this, and there were 3 people that became pre-diabetic. Yeah, so I'm not saying that we have everything in control, because this is 3 people out of 164, we have to figure it out. I'm just making the case that if you're saying that this kind of healthy behavior is a good idea, then you should also monitor, and not only by the scale, whether that single individual is doing well under that regime. Basically, the good news is that it was feasible for everybody, and they did lose weight, and it came with the reverse of a lot of complaints. People that sleep badly, they think that this is what they have to accept because they are of old age. It reversed. And we knew that already, but these elderly people didn't know. They're not that much aware that with a rather slight increase in metabolic health, they could actually decrease joint pain, they could increase their sleep quality, and so actually the quality of life for them increased. The nice thing is that a year later, they were still adhering to the regime because they did it as buddies. So these were either people with their friends or with their husband. Frankly, if I would do this with my husband, there would be no good about that, because I would want to adhere to it and he would quit within a week, I think. Yeah, so it has to be the right kind of motivating body. Now you realize that what we're doing is now we're checking all the metabolomics. So can you follow the different individuals in this intervention and see how their metabolic profile changed? And everywhere you see here this green sign, it is actually that those metabolites really changed into the more healthy metabolic profile. And so if you look into the metabolomics, we know already out of this study that by the DEXA scan we can see that the metabolomics profile is reporting specifically on your android fat, which is the worst fat you can have in your trunk. We know that it responds on the state of the gut microbiome. And Yotam is looking whether it's also responding to the state of the muscle in these people. And it does. So he's investigating, I think, so in 87 individuals, we had muscle biopsies before and after the intervention. And he has now looked into baseline and he can see that the different fiber types in muscle, which is of course a sign of your quality in histology experiments, that is actually already indicating how healthy the muscle is of an individual. And interestingly enough, we can now see that these signs of fiber, so the fiber typing, is actually associating to what we know about the body composition from DEXA, to the serum parameter, and also to metabolic parameters. And so we can now say, okay, we can read in, let's say, 5 metabolic parameters in blood, that really associates to the state of the muscle. And so we come to the situation where the metabolomics profile, it does give a kind of generic indication of your disease risk, but it would be multiple diseases. And it is influenced by your gut microbiome, by the quality of your fat, and also by the quality of your muscle. And so it is really reporting on your metabolic state. It is maybe, it may be not specific enough for a disease biomarker, but it may be useful in the clinic to kind of stratify elderly. I'm going to skip this. I'm just going to say that so if one thing is that we wanted to relate it to the biomarker, the other thing is that we want to understand the heterogeneity. How come that some people responded kind of so badly to something so healthy? And we knew that they were compliant. So that is why in all the tissues we have now RNA-seeked them, and we're now trying to investigate if we can find that some people, in a kind of personalized fashion, they're not responding well because at 2 different time points you see aberrant RNA being expressed in their tissues. Okay, so to summarize, the difficulty in a human study is that if you want to do big studies and have a good idea of the whole population, then you can only have blood. And you need more depth. If you want more depth, you need the tissues under the right conditions. Then you go to smaller numbers, and that is why it's so fantastic that with all this information about omics at the background, we can now do these experiments and get much more out of it than we used to do. And so this was a pilot study, and now we're gearing up to see what we can do in the clinic and whether we can make proper biomarkers for the clinicians out of this. Thank you very much. This is the group, and this is the funding.
Thank you so much. Very enthusiastic talk. Unfortunately, we are a little late.
Yeah,
so,
I know.
And 1:30, there will be a general discussion. So you save your questions for the speakers.
I'll be happy to answer.
Yes. So Eline, one more thanks.
Okay, thank you.
Okay, our last speaker is Professor Frank Madeo. It is a great pleasure to have him here. He's a dear friend and collaborator. So Frank graduated in biochemistry at the University of Tübingen in 1994. After receiving his PhD in biochemistry, he established his own group there, but then he moved to Karl Franz Science University in Graz in 2004, where he became chair professor. So his group has moved to Austria in 2004. During his PhD times, he discovered that yeast cells can undergo programmed cell death, or also known as apoptosis, and thereby challenged the textbook dogma that apoptosis is restricted only to multicellular organisms. This finding led to the initiation of new research field, including our own conference. So he's the founding father of the field in which I am, and he organizes very nice conferences every 1.5 years. In this field, we use yeast as a model not only for apoptosis, but also for neurodegeneration and cancer research. And you have seen some of the examples throughout the talks, for example, from several speakers today and yesterday also.
particularly our young audience. And we have the speakers of today on stage ready to answer any questions. So, but I think we'll begin with one of the questions that was delivered in written form.
Yep. This one is for Dr. Hood. In an age of P4 medical data, how do you think this data can be protected? How can it be protected, this data?
So I guess the question is whether once a lot of data is generated from you, you can protect that data from insurance companies, employers, families, and things like that. You know, I think that is the one absolutely critical issue in this age of big data in personalized medicine. In the US, we have two laws that let personalized data be protected. One is Obamacare, and the other is a law called GINA. But they're incomplete because long-term care, some other kinds of insurance, aren't covered with those. So when we have, for example, people participate in this group of 108, I had to say to them that there are some types of insurance that we cannot guarantee you have protection against, and so forth. So I think for discrimination, it's absolutely critical that countries pass laws that actually forbid any kind of discrimination. On the other side of it, the whole issue that we brought up with the 108 pioneers is that if we use your data de-anonymized, we'll be inventing the P4 medicine of the future that will transform the health of your kids and of your grandkids. And under those conditions, 96% of the 108 agreed to have their data used. So my argument is that a patient shouldn't own their data. Society has generated the resources that made it possible to create that data and come to provide you with much better health from it. So it is the individual's responsibility to be willing to allow society to use that data to invent the medicine of the future. On the other hand, it's society's responsibility to protect against discrimination and so forth.
Thank you. We'll take one more here and then we go to the audience.
Yes, wave your hand if you want to ask a question.
So this one is to Professor Madeo. Could you provide a simplified explanation of the connection between spermidine and autophagy?
A simplified explanation. So basically, spermidine is doing something with transcription. It's entering the nucleus where it changes the epigenetic feature. And this epigenetic feature then leads to a change in transcription. Basically, it enters the nucleus and it leads to more autophagy-related genes that are transcribed. So the whole protein apparatus that is required for autophagy induction, there will be just more there in the cell because more transcription for it is there. And so this is, to put it very simplified, how spermidine, in contrast to other things like rapamycin, induces autophagy.
Okay. Do we have any questions in the audience?
Should I? Yeah. Professor Madeo, you mentioned very briefly mental health, and I was wondering, stress and overstimulation from stimuli. Is there any research ongoing on the cause of decreased, especially value of life, because of this?
I didn't get that acoustically.
Is there any research that correlates stress with decreased value of life and also decreased life expectancy? And is it ongoing, that research, or neglected?
So the question, if I understood correctly, is if there is a connection between stress and life expectancy.
Yes.
Well, I mean, that's a tough issue because here you are at a border between psychology and aging research. So you have to ask very fundamentally, what is stress? You know? So is stress the external things that happen, or is it the answer in your body? You know, there are people who are stressed even if they would just have one simple task per day, and others who are, you know, doing million things are cool as ice. You know? So that's the first thing. If you say, we have to talk about internal stress, I think. And what has been shown is that people who are internally stressed have shorter telomeres. If that then leads to shorter lifespan, this is not clear. But probably something is happening to you when you age, when you are stressed all the time, yeah. But you know, it's stuff, there are not many studies on that. Walter, maybe you could comment on that.
In general, it is associated with a shorter lifespan, but these are not big effects. So people that have higher stress, for example, people in New York tend to live longer than everybody else, right? In New York City, right? So you could say that obviously it's not the most, stress is not the most important factor.
So we performed reverse studies to investigate whether people that are so long-lived, in long-lived families, if that goes along with a better resistance to stress, for example, or other personality traits. And it's for sure that people in these type of families, and also in general in the population, people that have very long-living parents, they have, let's say, more optimism. So that goes along with less stress. So in the same situation, you just don't experience things as stress. And then what is especially contributing to a longer life is the kind of optimism where, so you have different kinds, the optimism where every day is a new chance. And so, you know, that's also a survival mechanism for elderly people. They start losing friends and even children. And the more you have the ability to make something out of every day, which is a kind of active coping strategy, the better your chance to become a long-lived individual. And this type of psychological profile is really present in long-living families. Yeah.
Then, as a quick follow-up, does psychology and behavioral research not have a place within the aging research community?
So the question is whether it should have a place or whether it has?
As I assume it doesn't, but should it maybe?
Well, in human research, it certainly has a place. So people are having setups for stress research. You know, people have to give a lecture, for example, and start sweating. You didn't see that, but no. But so they put cameras on, and then they investigate the physiology of people, how they respond to stress, and how that relates to their genetic profile. GWAS research into which loci influence the personality traits. And so all those results are taken over also by the aging research. So for humans, there is definitely a connection there.
Thank you.
Okay, the microphone is here. Wave your hand so we see you. There.
Do you believe that biology is the key to stopping aging?
Is the key to delaying aging? Is that your question?
It's delaying it indefinitely, like, you know, not really immortality, but...
Well, I mean, let me ask. Obviously, yes, right? So we know that in something that I mentioned in my talk, I mean, a mouse lives 2 years and a person lives 50 times as much, right? So we already know that the genetics and the epigenetics and what I call the program, the longevity program, is in charge of the lifespan. So now there's some sharks that have been shown to live 400 years. So you see how the biology is completely in charge of the lifespan. That doesn't mean it's a guarantee, but certainly a very, very high chance that a person is going to make it to 80 years of age, and a mouse will never make it to 80 years of age, and a mouse will get cancer when it's 1.5 years old, and people will get it after 40 or 50.
I would certainly say biology is the key to aging as well. And I think in two different ways. One is the biology can be used to understand the fundamental process of aging, and we've heard some excellent lectures on that today. And those insights unquestionably are going to give us deep and I think very profound means to delay the aging kind of process. But in a second way, I think the biology is key too, and that is these dense, dynamic, personalized data clouds that I talked about discover for each individual fundamental flaws in their genetics or what their environment has done to them that for the most part are correctable. So you can use biology to correct the deficiencies your genetics plus environment have left you with. And these are obviously more general kinds of things, but in both regards, discovering the fundamental mechanisms and discovering what you're like now and how we can optimize your wellness and hence improve your ageability, I think are really key.
We have a question here regarding these dense dynamic personal data clouds, so we can maybe continue on that. What are some of the things you think are important to have in these dense dynamic personal data clouds, and why are these important?
Well, the things I described for you that we do have in the dense dynamic data clouds are, one, your complete human genome sequence, and that is your genetics. And there are hundreds of actionable possibilities within your genome, so that's one very important element. We also carry out every 3 months blood tests on proteins, on metabolites, and on clinical chemistries, and they give you, again, separate and powerful new views of your health and what it's all about. We're doing discovery science on the gut microbiome. That is the ratio of species of microbes that live in your gut. There are probably about 10 times as many microbes that live in your gut as there are cells in your body. About a third of the molecules in your blood come from the gut microbiome, so they utterly, profoundly influence our health. But we're in the earliest stages of discovering just how they do that. And then the other area is the data that come from the quantized self, assessing activity, assessing quality of sleep, pulse rate, blood pressure, those common kinds of things. We also have taken periodic questionnaires that have to do with mental health, and that also gets factored into this whole equation. But when you do that, when you mix it up and analyze it, there are an amazing number of actionable possibilities each individual can execute to improve their health, that is, to improve wellness and or avoid disease. And I'll just say the dense dynamic data clouds, if you count the number of bits of data, it's in the billions of data points. Most of that does, in fact, come from the genome. But I would say there's an enormous opportunity in the genome to discover many, many more actionable possibilities. And I think we'll see a transformation in the next 10 years or so in that regard. And I think it'll become much more permissible for society to use that information effectively to optimize health for the individual.
Right, question there?
I have a question for Mr. Mondeo. It was said yesterday that if you mix, you get the best result if you mix several drugs at once. Have there been any studies in mixing rapamycin and spermidine at the same time?
If I got that correctly, the question was, if we add spermidine and rapamycin at the same time, if you have an additional effect?
Yeah.
Well, that's a good question. We are currently testing that. We don't know yet, but what I can tell you is that we see additive effects if we combine fasting with spermidine. So here we clearly have additive effects.
That's nice. No.
Thanks. I'd just like to follow up. I've got two questions, if that's okay. One is about spermidine, and you mentioned that a big contribution of spermidine comes from the microbiome, comes from the gut bacteria. So, can you say a little bit more about that, please, and maybe its relationship to fasting?
Yeah, I mean, we know that when we analyze the spermidine that is finally coming up in the blood, we realize that it's 1/3 coming from the microbiome. We don't know what the relation to fasting is when it comes to the impact of the microbiome. But what we know is that if you fast, and this has been shown by a couple of groups, then the diversity of your gut microbiota is enhanced. Okay, so which is good. Having a monoculture, which you probably would somehow raise when you every day eat only sugar, then probably you are full of candida species and these kind of things, then that's bad. But how this relates to the amount of spermidine production, we don't know. It's a new research field and nobody has ever looked at that, but we should do, yeah.
Okay, thank you. And if I could ask the second question, it's about sleep. And because I was expecting to be hearing something about healthy aging and how we all need to have good sleep at night to help our bodies repair. So I was just wondering about, for all of your research, and the influence of sleep on the different mechanisms. If you can say a little bit about that, please.
I'll just comment a little bit the other way around, which is the work by Sachin Panda, looking at time-restricted feeding and sleep, right? So he published recently that people, in part because of this crazy idea that you should eat all the time, you know, now we hear like you eat 5-6 times a day, and so, and the consequence of that bad advice is being that people, as he's shown, are starting to eat 16 hours a day, 17 hours a day, right? So people were still eating at 12, 1 a.m., 2 a.m., etc. And then what he did, he took a group and restricted their feeding to about 10 hours a day, and that really made big improvements in their metabolic profile, but also their sleep, their ability to sleep well. So I think that going back to some of these old ideas that we see in the centenarian population in the blue zones, where they eat within maybe 10 to 12 hours a day, say 8 a.m. to 8 p.m. It's a good idea also for the sleep, and also, obviously sleep is very important for longevity.
Maybe as an addendum, and this is also true for model organisms, and this has been shown by Linda Partridge. So if you overstimulate the insulin system in flies by too much feeding, for example, they sleep bad. If you take out the insulin signaling, they sleep quite well. They don't have as much sleep fragmentation then. So that seems to be true for model organisms and for humans.
You know, I'll raise just a point of skepticism. I don't think very good experiments have really been done on sleep and its beneficiality for health and so forth. There are a lot of really productive individuals over very long periods of time that survive perfectly well on 3 to 4 hours of sleep a night. So, I mean, in principle, it sounds like we should all get more sleep than we do. In practice, I don't think there are very convincing experiments I've seen out there that demonstrate that that really is true.
Maybe having said, I agree on that, but on the other point, for example, we had some, and others did too, interventions with either only exercise, more exercise, or exercise and nutrition in elderly. So all the time I'm talking about 65-year-olds, 70-year-olds, and we recorded their sleep, and you always have a part of the people that are bad sleepers, so they have really very poor patterns of sleep. And those definitely improve by exercising. So whether, and also if you do not do anything about nutrition, but you improve the exercising, right? So, but that could be another part. So these people are really complaining, so they're not, so all of us that might sleep 5 hours a night, we're not complaining because we do well during the day. But these are really people that have disturbed sleeping patterns when they grow older, and they suffer from it, and that part becomes improved by exercise. There's a lot of literature on that.
Right. Next question.
I can speak. Yes. So my question would be for Professor Slagboom. So now we are developing like new scores about metabolism syndrome, you said, and I was thinking about the impact on the clinical diagnostic because the clinical diagnostic, they are old-fashioned. They were made without this knowledge, and should we speak now differently from the diseases, or will it have such impact that on the clinic that or should we think in terms of more in system biology, or is it?
What we have now in terms of metabolomics and the ones I showed, those would not be so. So, for example, what the clinician needs is a biomarker that says something about an individual. You know, the individual risk, not the group risk. And any biomarker needs to give information on that part to change the diagnosis and change the treatment. But having said that, the biomarkers that are there are usually not working at 70 years or older, right? Nobody uses the Framingham risk score to figure out the risk of a heart attack of a 70-year-old unless you have all the signs that this person really is like a 50-year-old, because then they will treat him like a 50-year-old. Mm-hmm. So our starting point would be that as the metabolomics and the proteomics can improve, be more precise, standardized, being able to do it on thousands of people in the clinic, and also affordable. You know, if that was there already in practice, then we could start using that in the clinic, and first, of course, testing it, and then figuring out whether that helps in the regular treatment of elderly people in the clinic. So what we're doing at the moment is to motivate all the clinicians to at least start collecting the right material on all the patients in the clinic, because that's what epidemiological studies do, but clinical studies do not do that. I mean, the studies do it, but not the regular treatment of consecutive series of patients. Mm-hmm. So to be prepared for the best biomarkers to come, that is something that we have now started to do. And you might say, on the side, what you're already trying is whether those generic markers that are not personalized enough, if they could at least give some stratification in these individuals to show, you know, which people are at higher risk of sarcopenia, for example. And that is something they also need. But then you accept that the biomarker says something about the group and less so about the individual.
Mm-hmm. Thank you.
You want to add something to that?
I have a second question. It was about fasting. The effect, it's not, it's for Professor Walter Longo, I think. It was about fasting, so the beneficial effect is decreasing after aging. There was shown on with aging, that was shown on the graph. This means that many patients which would have a cancer, they cannot use this technique to improve the results of their treatment, or what do you think about this?
Yeah. So those are chronic studies, right? In mice, and we haven't done any in people. I mean, we've done it in people, but not in the very old. Up to age 70, people have no problems, right? But in mice, if you do it from middle age and you do it twice a month all the time, then once they get to very old age, they're bothered by it. And but I think if you're asking about a cancer patient, that usually you're talking about 4 or 6 cycles of the chemotherapy or other treatments, and other treatments, new treatments, are much less toxic. So we have seen very little evidence that that's a problem. Now, if you're talking about 85-year-old, it could be very cachectic, so somebody that is 85 and might have prostate cancer and has lost a lot of weight and a lot of muscle mass, yes, that's definitely somebody that the oncologist will probably refuse to put on a fasting mimicking diet. Keep in mind that we don't do fasting anymore. It's only fasting mimicking diet, which have relatively high caloric intake.
Yeah. Thank you.
Excuse me. I have one more question here. It's about spermidine again. Is it possible there might be some substances like spermidine, perhaps even more effective for its use, or do you think this is a unique substance?
Yes, I actually think there might be some substances out there that might be even more effective, because we didn't find spermidine in a systematic screen, but we just bumped into it by chance. Okay, so we now set up a systematic screen and compared spermidine to thousands of other substances that are plant-derived, because we believe that there is a long-lasting and long-standing co-evolution for millions and millions of years between plants and humans, and this is why probably most of the blockbusters in medicine still come from plants. And I think, yeah, there might be substances there that might even be more effective. We are searching for them. We haven't found them yet, but it's a question of probability. If you by chance find something, you probably have not found the best thing. If you have a real systematic screen, you probably do better. And just one sentence why I believe in plant-derived substances. Isn't it crazy that there is a plant growing on the fields, which is poppy, that produces a molecule that exactly fits into a receptor in our brain, which is opiate, which makes us happy. And this shows how potent this co-evolution through millions and millions of years have acted. And this is why I think also the plant-derived substances are so potent. And this is the direction we look now in.
Yes, I have a question for Dr. Hood regarding Alzheimer's. You said that if you could detect it at an early state, you could revert it. Could you tell us a little bit more about that process?
He's asking about the Bredesen study and reversion of Alzheimer's disease.
So what Dale Bredesen at UCLA has done is taken a systems approach to Alzheimer's and looked at compounds that influence one way or another synaptic connections, either their proliferation and or their loss. And he's put together, as I said, this regimen of 36 different things. It includes supplements, it includes drugs, it includes activity, it includes some aspects of diet and so forth. And he's tested this on about 125 individuals that are in the earliest stage of Alzheimer's. And what he's been able to demonstrate over about a period of a year is that of the order of 75–80% of these people have reversed back to a normal phenotype. And one of the really critical questions is the 36-point regimen is really an exhaustive kind of thing to ask the patient to carry out. What would be ideal is if we could take each individual and determine what were really the key elements in those 36 and narrow it down to 10, to say something like that. He hasn't done control studies, so we're actually going to set up at Providence a study on several hundred early stage Alzheimer's patients, take them through this initiative against controls and do it in such a way that once we reverse things, we can think about beginning to test which combinations of the 36 actually will keep it there. One of the big questions is whether you need to continue this regimen to ensure the reversal or not. And that's an open question at this point in time. My guess is you'd actually have to continue some aspect of it because, again, a lot of these people are at fairly high risk for Alzheimer's. So that's the essence of what we're going to do. In the second study, where we hope to be able to prevent it, we'll actually take a couple of hundred individuals that are at the upper 10% of susceptibility to Alzheimer's and follow them with the regimen before they ever transfer over to early stage Alzheimer's and see whether in these people you can actually prevent that transition. And these will be people in their 50s and 60s, so they'll be closer to that transition process and so forth. And I think these will both be really informative studies that could fundamentally change our whole attitude of that whole neurodegenerative disease. And of course, what's exciting is you can think about applying it to the other common human neurodegenerative diseases as well, Parkinson's and so forth. So we're just beginning those studies now.
Okay, we have a question in the back, and then here, and then.
Yeah, so I'm wondering, like, a lot of you talked about yeast cells, how you test a lot of things on yeast cells, and like blocking a protein, see what happens. But I'm wondering why we haven't heard a lot about HeLa cells, because they should mimic human cells even more, seeing as they are like human. And why we haven't heard about it if you do that.
I think if I understood right, the question was why are we, instead of using yeast, not using human cells like HeLa cells, you mean?
Yeah.
Okay. I mean, there are many questions, there are many answers to that. You know, a cellular system in cell culture is per definition an artificial system. And there has been published a couple of results on human cell culture system that just had nothing to do with real biology. I give you one example. There are many data out there on neuronal cell cultures, and they're just wrong, because you cannot put a three-dimensional structure like a brain and put it into 2 dimensions, and then these neurons are firing, for example, in a frequency which is totally unphysiological, which has never been seen in a brain. So that's first. Second, with yeast, you can do fantastic genetic screens and tricks and learn really new pathways, as for example, Walter did with some mutagenesis and so on. Third, yeast has been proven successful. So I would say 50% of the things we know today in human aging came from very basic yeast studies. So for example, the sirtuins, they have been found in a yeast screen. And they are now known to have a lot to do with age-associated diseases in higher organisms. So there are many, many reasons for that. Walter, do you want to add something?
Yeah, I would say that for those of you that are going to go into science, in the model organism is obviously very important for what Frank just said, but also the cells are very important. So I think you want to do parallel studies primarily with primary lines, right? So not transform lines. So the transform lines are the ones that cancer, the immortal lines. So those are much less representative of what's going on in vivo. But I think if you can do yeast or C. elegans or flies, and then at the same time go take human cells or mouse cells in vitro, and then maybe work in mice, I mean, this is what a lot of laboratories are now starting to do. So I think the human cells or mammalian cells is very important. We always do it in parallel with yeast, and we learn a lot from it. But yeah, I agree with Frank that when you're looking at what is a gene that extends lifespan, the mammalian cell in culture is going to be a very poor model to give you an answer.
Maybe. I do not completely agree. I think there's quite a distance between yeast and humans. And I think for human disease, the cell biology, and especially with the iPSC technology, where you take fibroblasts from individuals and you de-differentiate them and differentiate them into different cells. I mean, it's never exactly the same as what the cell would be in the body, but if you want to investigate functionally, you know, you have found, with all the sequencing, you have found a variant, and you have the suspicion that this variant is important for human disease, but from an epidemiological point of view or a genetic point of view, you can't get your evidence straight, because there are just not enough individuals with this rare variant
around to investigate it. Then you go with the variant to cells and specific stem cell population. This is what we did for osteoarthritis. And then you get a lot of answers. So personally, I think for human research, the cell biology and also single cell technology is vital.
Yeah, I was going to make exactly the same point. We've done some studies recently where we've taken iPS cells, induced pluripotential cells, that you can stimulate to differentiate to cardiomyocytes, the fundamental cells in the heart. And this is a process that takes a week plus or something like that. And what we've done for each day during that week is to take 1,000 single cells and quantify their RNAs. And we've been able to demonstrate in that differentiation process, one, it mimics perfectly what we've learned about cardiomyocyte differentiation in the organ itself. But two, if you do it from the single cell point of view, you learn all sorts of new biology about how the process is done and the complex interactions between different cells and things like this. So I think we have the ability now to create very reliable copies of virtually any major cell type, and then to be able to study it in vitro. But I'll go back and say, I think the power of yeast genetics is absolutely staggering because of the genetics and the high throughput screening and all of the other tricks that we've learned over the years. And of course, what is absolutely fundamental is that the core biology in yeast is really replicated in humans to a remarkable degree. Even the molecules in many cases are direct copies. They're directly orthologous one to another. So I think the model organism state lets us do things in a way we can't at all do it with human cells. And I think being able to move back and forth from the model organisms to the human cells is really the key to doing it most successfully.
I think it sounded like a disagreement, but actually I think we all agreed, right? So you need to work in parallel with all the systems. This old idea that you can just stick with one system is an old idea, right? You really, if you want to solve a problem, you have to do the yeast or the worm or the flies, then the mammalian cells, then the mouse, and then the actual human studies, right? So all of the above and the omics approach, right? So all of those are really required to get real answers.
Which actually is the subtitle of this symposium. We are running out of time. We have time for a question now.
A question which might be a bit different, something that I came to think of during the first talk. With all of these, especially the clinical trials that you've talked about, we have a bunch of diseases which I think most people would agree are quite terrible, like Alzheimer's and different cancers. How would you ever get people to participate in them in the control group? I really don't understand. And of course, that's quite important, and especially since I think most people in here at least are thinking about going into research. What would you do?
Well, these are standard. There is a randomization process, so they don't know they're going to be in the control group. They're recruited, and then they're assigned to the control group. Sometimes you do what we did is crossover, so they're in the control group, and then they're crossed over to the experimental group. That's much easier. When people know that that's the nature of the trial, that's much easier to do it.
Yeah, I was going to say the same thing. With the Alzheimer's, we're going to do the crossover because it would be unfair to assign really high-risk people forever to the control group. So that crossover ability is really important.
Thank you.
So one question from the box. It's for Leroy. I'm going to shorten this. Are there any big cons with personalized medicine? And that could be, for instance, if it's too expensive or something like that. Yeah.
The question of the expense of personalized medicine, I think, is a really fair one. But there are many answers to that question to show in the end it's going to be dramatically less costly for healthcare than is the medicine we practice today. So number one, I would give you the example of the 10 drugs I showed you where the successful populations varied from 25% to about 4%. So you're giving drugs to 96% of the people. The drugs don't do them any good, and in fact, in many cases, they do them harm. And in fact, in all cases, if they're expensive drugs, they cost the healthcare system and the individual a lot of money. Personalized medicine is really going to give us the ability to stratify populations, to identify responders from non-responders. I think another area that we're just beginning to realize the magnitude of the savings is if we can prevent people from getting sick using this dense dynamic personal data cloud approach, and or if we can identify sick people and reverse them back to normal, we can again end up saving society enormous amounts of money. We had one individual in the 108 pioneers that came in complaining of an arthritis in his feet that had been occurring for the last couple of years. His physician absolutely couldn't figure it out at all. And when we analyzed his transferrin, it was blazingly high. That's an indication of how much iron you have in your blood. And when we analyzed his genetics, he was homozygous for defects in the gene that cause hemochromatosis. And hemochromatosis is a disease that starts initially by arthritis. It attacks the beta cells in the pancreas. Then it fibroses the liver, and finally, it causes cardiac decompensation. Usually people come in with cardiac decompensation, and by that time, they're chronic, costly individuals for the rest of their lifetime, costing the system hundreds of thousands of dollars to manage and process. So it doesn't take very many people per 100 of that type to really end up saving the healthcare system enormous amounts of money. And I think after we've done the control studies at Providence of the 2,000 employees that will experience scientific wellness against the appropriate controls, we'll be able to calculate the economics. So I think the idea of personal medicine really being expensive is a false one when you take into account the enormous savings it's going to generate.
Okay. I
Okay.
think that will be the last question. Thank you so much for your answers, and thank you for your questions.
I think you should give yourself an applause.
Oh yeah.
So you should stay because we're going to have a panel discussion. That's the next item. You stay. And we add some people on stage. Yeah.
I could just introduce myself. I could introduce myself. I don't need a big introduction.
Okay, welcome to the last session, which is going to be the panel discussion. So it is a discussion that will be involving all of the speakers from the program. Some of them, as you are noticing, are missing because they had to leave early, unfortunately, to catch their flights. And the person who is going to moderate the discussion is Mene Panagoulos. He's a neuroscientist best known for his work on the discovery and development of novel drugs to treat Alzheimer's disease. Dr. Panagoulos commenced his scientific career with an undergraduate study in biochemistry from Imperial College in London, and he did his PhD at the Institute of Neurology, also in London. He did his postdoc training at Mount Sinai Medical Center in New York. Menalis Panagoulos was the vice president of neuroscience research at White, a leading pharmaceutical company, and was responsible for overseeing the transition of more than 20 novel drug candidates into clinical development for a number of neurological and psychiatric diseases, including Alzheimer's, stroke, schizophrenia, and pain. He previously served as a group director and head of neurodegenerative research at GlaxoSmithKline in United Kingdom and held positions with SmithKline Beckman, Janssen Pharmaceutica, and Bristol Myers Squibb. In 2010, Dr. Panagoulos was appointed head of Innovative Medicines Unit of AstraZeneca. Dr. Panagoulos is also an adjunct professor at neuroscience at the University of Pennsylvania and visiting professor at King's College in London. He's on boards of several scientific journals, universities, and funding agencies. He's a member of the American Society for Neuroscience, British Pharmacological Society, and associate of the Royal College of Science. He has also written several books and more than 100 scientific papers. Welcome, Mene, and we look forward to hear from you.
And that's very I don't keep up with Wikipedia, and that's very out of date, unfortunately, but never mind. I'll go back and correct it on the plane on the way home. So my role at AstraZeneca is to run research and development across all of our therapy areas. So to take the scientific ideas, many of which you've heard today over the past 2 days, and try and turn that into something that become a therapy for patients that will make a difference to their lives. I just want to, if you just put the first slide up, I want to just spend 5 minutes just talking a little bit about some of the lessons that we've learned about drug discovery, because we've done quite a lot of work to try and understand what's the difference between good drug discovery and bad drug discovery, and hopefully that can lead into some of the conversations and discussions we have later. And when I joined the company in 2010, we were very much driven by the volume of what we did, not by the quality of what we did. So how many drug projects we had, how many candidates we had, how many new targets we were starting up, versus how strongly did we understand the biology validating that scientific hypothesis. So when I joined, we actually killed more than half of the programs in our organization, and we did a piece of research to try and understand what separated successful drug discovery programs from unsuccessful ones. What was the type of data that we had, what decisions did we make, and what was the consequence. And we came up with a framework, and this is actually data that's been repeated now by quite a few other companies, and we call it the 5R framework. And it's not so much that it's a 5 Rs or a framework, but it's more the types of things we have to think about when we think about starting a drug discovery program that ultimately we want to take into man, which is a very serious thing to do, and test it to prove or disprove a hypothesis that we think will have a benefit ultimately to patients. So the first R is the right target. How well do you really understand the biology? How well genetically validated is it? How do you take data from humans back into your preclinical models and back translate? And how do you continue to increase the confidence of that biology as you move through your program? You don't just stop, but you keep on doing that research, keep on trying to understand, to disprove your scientific hypothesis, to demonstrate that you can actually increase your confidence. Second R is about right tissue. It's great to talk about molecules and antibodies and peptides and things that work in yeast and flies and mice, but ultimately you've got to be able to demonstrate that whatever it is you're trying to hit or target, that your molecule gets into the right organ, to the right enzyme or receptor, and blocks it or activates it for the right amount of time. So if you have, for example, a drug for schizophrenia, you need to be able to demonstrate that your drug for schizophrenia gets into the brain, hits the right part of the brain, and actually blocks or activates that part of the brain for the appropriate amount of time. If you don't have that, if your drug fails, you don't know if it failed because your scientific hypothesis was wrong or because your molecule was lousy and didn't actually engage the target. So having ways of demonstrating that you're engaging your target is very, very important. Right safety. We've heard actually during the course of the past 2 days around the challenges, for example, with TOR1 inhibitors, drugs like everolimus. Some drugs are not very nice and very safe. So understanding your safety signals early, and then making sure that those safety signals are thought about appropriately, so that you're not putting toxic molecules into people where they won't be tolerated, where the risk-benefit is not adequate, is a very, very important thing. And you can weed a lot of bad molecules out very early on in the drug discovery process before you spend a lot of money in the clinic. Right patients, we've heard even in the course of discussion in the first panel session, the importance of defining the patient population that is most likely to respond to your therapy. Do your experiment in that patient population first, because if it doesn't work in that patient population, sure as hell isn't going to work in a broader patient population. And the more defined that patient population is, the more likely it is that your therapy ultimately will work, because it also probably means that you better understand the disease and what's driving the disease in that particular population. And then finally, right commercial. And by right commercial, I don't mean is it going to be a billion-dollar drug. I mean understand what the size of the opportunity is, so you can then spend the appropriate amount of money in research and development to make it a good return on investment. And that becomes something, again, that's very important as you think about commercializing things. It's not whether it's a big drug or a small drug, but if it's something that's in a very niche population, a very rare disease, think about how you do the appropriate studies preclinically and clinically to ultimately make it affordable when it gets, hopefully, to being commercialized. So that kind of, by following that framework, we've actually seen our success rates go up dramatically in terms of our ability to take ideas from the lab into the clinic and ultimately to launch drugs. We've gone from being one of the least productive pharmaceutical companies to now, I think, one of the most productive. We're still very much on a journey. And there are many other things that we can talk about, and maybe we can talk about with the panel as well, that we're trying to do around being more open, more collaborative, using the brain of the world rather than the brain of AstraZeneca scientists to really try and tap into ideas, new science, new innovations to really help us tackle some very, very difficult diseases. So let me open the panel discussion. Obviously, the topic of the conference has been around healthy aging. We've seen some amazing data, and we've seen data in yeast and in mice and in flies. And we've seen quite a few graphs where we look at improving longevity of whatever organism it is that we're studying. One of the questions I have for you is, how do we actually turn that into therapeutics? So what's a disease indication? Obviously, we've heard some data that we can improve, for example, sensitivity to chemotherapeutics, or we can improve outcome in Alzheimer's, but aging in itself is quite a nebulous topic. If I think about it from a drug discovery perspective, what are the diseases that you think are best suited to aging research? What are the types of endpoints that you think we should be looking at or using? Should we be doing prevention trials or progression trials? So why don't we open it up and start with Ellen? Ellen? Yeah, what are your thoughts in terms of how you turn some of this fantastic aging science into therapeutics and ultimately clinical trials that you can use to come up with a medicine?
You asked me this question, and I haven't thought about it, but what I can imagine is that if we see certain molecular processes that go up in many of these lifespan beneficial or lifespan prolonging treatments like autophagy, so you could take that as an endpoint, so the molecular pathway rather than a disease perhaps. So that's what comes up in mind immediately.
Other thoughts? I want to go through the autophagy. I think would be quite a difficult endpoint for a regulator to approve. So I think, again, it's not an easy question to answer, but what are the types of things that you think you would do to really take aging science and actually put it into practice in the clinic?
I think one of the first things you'd have to do for aging is create metrics that we can use to actually assess whether you've blocked, whether you've reversed, transformed the aging process. And I think one really interesting metric is the quantification of your physiologic age as opposed to your chronologic age. And actually there are people who've argued that you can use DNA methylation to begin to make determinations like that. The arguments aren't convincing, but until you have metrics for aging, it's really even going to be hard to think about how you could possibly set up the assays that could look at drugs that could change the whole process. I think the second thing that's really interesting is in either human cellular systems or even the model organisms, can we get reasonable proxies, quantified proxies for aging we could use to come to better understand the phenomenon and so forth. So I think there are a lot of general things you can do along the wellness category that I discussed that I think in time will really inhibit the aging process in a striking way. But they aren't taking us toward drugs. They're really taking us toward known actionable possibilities at this point in time.
Leroy, just before you, Leroy, how would you make it broader? So let's say you're obviously setting up a platform where you're looking at healthy aging, trying to move it along a healthy path, or if it's become unhealthy, change its course. How would that become broader in its utility so that it becomes used globally, not just in the US, but Europe, emerging,
So we're actually already negotiating with 2 different EU countries about the possibility of bringing a substantial scientific wellness study to those countries. And I think your point is really a good one. We want to take into account the variability of human polymorphisms in different populations. We're talking with people in China about exactly the same kind of thing. So if we can begin by getting in Asia, in Singapore, India, those are 3 countries we've talked to, and several here in the EU, I think we can begin to collect the genetic variability that will say, how variable is that wellness response in different populations?
Walter, you go.
Yes, I was going to say that the FDA actually is putting some effort towards this, and near Barzillai in the US has already assembled a program to test metformin, and the idea is not to test metformin to delay aging, although there is the secondary idea, but first they're going to test it on people at high risk for cardiovascular disease, high metabolic syndrome, etc. So test it in them, and then 10 years down the road, if it turns out that it's clearly beneficial for even the people that might have very low or lower risk for these diseases, then I think the idea would be to introduce it or maybe test it further for aging. We're doing the same thing with the fasting mimicking diet. So we're looking at cholesterol, and we're looking at hypertension, et cetera, et cetera. Obviously, at some point, now we've probably had over 1,000 people that have done this. At some point, we'll have enough data to say it's affecting healthy people. They might just have, let's say, fasting glucose of 99, right? And it brings it down to 90, right? So at some point, you can say this is clearly also by using papers by many. If you have 20 risk factors or factors or biomarkers associated with aging, and they're all improved or generally improved, you can say this is probably affecting the aging process and begin to recommend it for that purpose.
And do you think diet will be something that, I mean, I know I'm crap at diets, pardon my French, in terms of following them and keeping them, do you think, as you generate more and more data with dieting and fasting, that will be something that would become common practice, or will you ultimately have to replace it with something else because people, unfortunately, generally aren't very good at following their doctor's advice? They're not very good at taking prescription drugs either, I have to say. But how would you make it into something that really would have a global impact in terms of health? Something like dieting, for example, or fasting.
Yeah, I think you're going to get both, right? So there's going to be a lot of people that want the drug, and they want the easy way out. But I think we're also seeing that there's a lot of people that want the safer intervention. These are periodic things that we believe, for example, people do once every 3 months for 5 days, right? So these are not, I mean, the idea is not to have calorie restriction like the poor monkeys in Wisconsin and the NIA that have been put on this thing for 30 years with, limited results, but to have something that you can, go to the doctor and he prescribes it to you, you do it once every 3 months, and then you go back to what it is that you do. And we saw, surprisingly, the cardiologists, endocrinologists, gynecologists were actually very favorable. I mean, not everybody. Let's say 30% said, this is something I really like, because I see that it's more comprehensive than a drug. Very unlikely that 10 years on the road I'm going to find out that it does, testicular degeneration like for rapamycin, right? So yeah, so I think both. Right?
Do you think that will be the next increment in terms of? Do you think caloric restriction will be the next thing that kind of you see in terms of the research having an impact on people's outcome? Do you think that will be the next innovation in terms of how it gets used?
I really see, we see in many countries now we're starting to introduce this, and we think that there's going to be, again, a percentage of doctors that just love this idea.
Yeah.
And they say, I'm much rather go with this and try to fix the problem than put a Band-Aid on it and delay the problem. So we'll see, but yeah.
I think your point of how do you get people to continue on this long term is really a critical one. And the approach that we've taken with scientific wellness is to use the coaches to ensure that people stay on long term. And in the first year, as we're turning over at Airvale, the re-up rate for the second year is fantastically high. And almost always people cite it's the coaches and their helpfulness and their interactions. And we have the feeling that we have a cadre of people that will be continuing. I mean, we're making the argument this should be a lifetime, but the data are going to be incredibly valuable. And in a sense, it grows with an exponential increase in its value as the length of the time expands out and everything. And again, the actionable possibilities, nutrient changes, things that can change inflammation, things that will change prediabetes, all of these things we're going to have in time, very large scale N of 1 studies that can be aggregated.
Yeah, please.
I think it's also very important what kind of organizational structure you have around it. So, for example, in Leiden, we have a lot of discussion, like you cannot talk about diabetes, it's either the younger diabetics or the older diabetics. And when you're talking about the older diabetics and you first want to keep everybody, not to use medication in the first place, but first preventative things to do, then what it takes is that you target the right group of people, that the general practitioners already start targeting that, and there's a direct line from general practitioners into the hospital with the conviction or the people being convinced that you have a certain order of events. First, you try really to prevent, or maybe to do, better nutritional intake. It needs to be monitored well, and that is, at least for the Leiden area, there is where this focus on metabolic health comes from. Are there ways to improve that in the first place? And so, if you all start to use the right material and markers, then the next phase is. If the hospital then starts treating individuals, then they should also be eager to discriminate different groups of people. So there will always be people that need the worst medication almost instantly, and others do not. So the best future world would actually be where this hospital is also in an environment that where people are not so triggered by commercials to always behave badly. Right? So there's a lot of discussion ongoing that you should actually also have commercial partners and stimulate economic aspects that are more focused on the healthy trajectory than on the hospital part and the pharmacology. And I think it takes more than just, us testing medication or clinicians monitoring patients. Also is this environment where, because people can almost not change their behavior, they have to change as a whole family. And in that sense, our experience is that the whole familial effect, for example, for metabolic syndrome is vital. But if you go to the older ages, people have motivations of their own. You can much better motivate 70-year-old people into a preventative health change. So the whole organization needs to be focused on that. Yeah.
I'll have a go and then we'll change topic a little bit.
So when it comes to supplements, I would quite naively say that science will find its way. There are currently like 4 or 5 supplements in the discussion of being age preventative, and clinical trials are running with all of them. And for sure, some will have success and some won't. And the ones that are successful, that other scientists will jump on that and continue, you know. And one day, it's clear that, for example, supplementing, let's say, rapamycin or whatever, is beneficial, and then everything will go automatically.
Yeah, the only, just to push a little bit, the only challenge I would say is that generally speaking, and I'm generalizing now, most of the trials that have been done based on post hoc data, like for example with NSAIDs in Alzheimer's disease or various supplements, generally in AD, which they've almost all failed. In fact, they've all failed, not they've almost all failed. And so there's something, I think, more challenging when you start to get into a clinically controlled environment that makes seeing efficacy a little bit more difficult, which is why the endpoints and the disease and the patient population you go into are so important. And that becomes, I think, one of the challenges why I'm asking the question about where do you think you have the best chance of having an impact in terms of seeing a benefit. When we do, you know, there's lots of data on statins and NSAIDs having a very positive protective effect on Alzheimer's disease. Every controlled trial has failed, including PPARs and everything else, and hundreds of millions of dollars have been spent doing those studies both by pharma and academia. So I think it's a little bit, you know, sounds nice, you know, some of them will work, but the question is which is the patient population that you go after where it has the best chance of working to try and make that investment worthwhile. Tough question. Let me ask a different thing. So we've got a lot of budding scientists in the audience. If you look 5 years out, 10 years out, what technologies do you think are going to transform your research and what you work on will have the biggest impact in terms of moving the field forwards? You can go down the line, you can go up the line. Thomas, why don't you have it? Do you want to have a go?
In terms of technology, I don't know. I think there's been a lot of talk about new technology, systems biology, genome-wide approaches and so on, and those are great.
Yeah.
I don't think we should forget about how important the reductionist approach is still. So we need to have that in parallel with all the new techniques and the genomic and metabolomic techniques and what have you. So I'm sure there'll be technical advances that will be really important. But one of the things I think might be a golden age, and that we're entering that age, is a golden age of new cell biology, new organism biology, which in part is due to aging research. Because traditionally, a cell biologist, as an example, have looked at one process, or even one molecule, one organelle at a time. And what aging research is bringing to the table is that they're all connected. Everything is interconnected, and you see that in aging research because you see that one thing is breaking down, and sequentially, like a domino effect, other things are breaking down because they communicate and there's an interdependence. And the result from that is that aging research gives us a new knowledge about how the cell is organized, how the organism is organized. And on the one hand, that might sound negative because then you say, okay, so there's a domino effect, the mitochondria breaks down and shit happens sequentially and everything breaks down and you're dead. But the other thing we've learned from aging research, and I think that will be more and more important and maybe also in drug development in the future, is the adaptability of the system. So you very often see that something is breaking down, or you deliberately knock down a gene and you reduce mitochondrial function. And there's no way they can repair for that. But they have compensatory mechanism of responding, sensing the defect and responding in very unusual ways that we didn't know. And sometimes they overcompensate. They actually have lifespan extension. It's a classical example when you reduce mitochondrial respiration. In many organisms, you have lifespan extension because of overcompensatory mechanisms. So I guess maybe in the future, you might not even need to attack the root cause of what's going on, but you may attack the compensatory mechanisms that actually could overcompensate for something that, you know, will happen anyways.
Linda?
I think a lot of the invention, as opposed to bits of kit, is going to come from data analysis. I think it's a bit like trying to understand how the brain works. A lot of the systems biology data, huge data sets collected at different levels from different tissues, sometimes with some systemic measurements as well. And understanding what's going on and how it all fits together, I think requires a whole cadre of, to some extent, newly trained people who really understand biology and biochemistry, but are also crack computer modelers. And I think that there's going to be huge expansion in that whole area of data analysis. But if you don't mind, I'd like to backtrack to the previous conversation just briefly, which is, I think, what's come clearly out of the aging research is that you're talking about drug development. A lot of it is going to be preventative measures, and preventative measures only work if they're preventative. And if you try and implement them too late in the disease etiology, then it isn't going to work. And we have an almost classic case in the lab at the moment with mice where we have a drug from a company against a target. I won't name either of them. But it failed in preclinical trials in the company because it was being developed as an anti-cancer chemotherapeutic, and the way that they tested it was by introducing already tumors into mice, which were compromised in their immune defense, because that's a classic method of testing for anti-tumor drugs. And they found that they didn't stop the growth of the tumor. But if we give it to mice before they have tumors, they don't get them. So I think we do need a slight head change in the way that we think about all of this. And actually, I would say cancer prevention is an area that we should look at a lot more carefully, because we already know that rapamycin, for instance, prevents cancer in mice, but people seem somehow to regard that, to me, major discovery as rather trivial because they're interested in aging. But the mice don't get cancer, for goodness sake. And if we could prevent people from getting cancer with early treatment, it would be wonderful, wouldn't it? So to answer your question, get a genetically at-risk group for a particular cancer and see if it stops it.
Yeah, it's a great.
That would be my best bet.
Great point. And of course, hopefully with prevention, you also need lower doses, which are better tolerated as well, which makes a difference. Technologies, any other comments on technologies or what you think the future holds?
I don't know if this has been mentioned, but I think the development of iPS cell technology and organoid technology in combination with CRISPR technology will allow you to use patient-derived material, and I think that will be very important.
Yes. Yeah, I agree completely.
Yeah, I would agree. I think three-dimensional tissue organization chips are really going to be transformational for reproducing real biology. I'd say a second area that's really going to be transformational is, again, this third-generation sequencing, where we'll be able to sequence whole genomes from a single cell, and that really opens up new kinds of possibilities. A third area is the area of making reagents to replace antibodies, both as diagnostic and as therapeutic reagents. And of course, the limitations of antibodies are that, one, they have enormous cross-reactivity. Two, they're enormously unstable. You never know when they're really going to work or not. And three, they're not easy to produce. They aren't digital in any sense of the word. So recently, Jim Heath at Caltech has developed a technology that has the ability to take circular D-amino acid 5-mers and use them as monomeric subunits, assembling them on the protein you'd like to make the capture agent for, and using click chemistry to join them in the appropriate three-dimensional orientation to make them dimers or trimers or whatever you'd like. These have enormously high affinity. You can essentially eliminate cross-reactivities by targeting them against computationally chosen epitopes in the protein. And they're digital in the sense that once you know, say, the two monomers plus the nature of the coupling agent, you can put that in vitro and with copper catalysis assemble a pound of it. So these are reagents you can put in an envelope and send to Europe. Their affinities will be unchanged. And these are reagents that have the possibility of in vivo or in vitro diagnostics, but as well, they're going to be fantastic therapeutic reagents. So I would say another area that's going to be really important is in vivo molecular imaging, being able to look in the organism at molecular interactions and reactions exactly where in the relevant position that you'd like to see them and so forth. So I think those are among technologies that I would also say the analytic tools are utterly going to be transformational for dealing with these complex data sets and so forth. And the future is being invented there. Talk about wonderful kinds of opportunities. And the other area is, I think, the use of microfluidics and nanotechnology to miniaturize and parallelize and render far less expensive all of the assays that we now struggle with that I talked about in scientific wellness.
Can I add something to it? Because I agree with all the former speakers, but I think it's also vital that if you have all this technology and you learn so much about the personalized medicine, is that you also combine it with physiology. So, for example, an enormous amount of the omics in our populations has been collected in fasted samples, right? We're doing it at every level of organization, but that's actually not the most interesting state of a human being. You know, so you try to do things in cells, but actually you should have more physiology in humans and then measure all the omics that you can get, and then have the right computational biology. I think that is going to maybe a big help.
Frank?
So if in 5 to 10 years from now we really would have a supplement that would delay the onset of aging or age-associated diseases, then I think we need another form of technology, because then there will be a big run on that substance, and we need terribly methods then to produce it cheap, because everybody in the world would want to have it. So we really have to figure out then a way to make it available to everybody, and that's a big challenge for industry, I think, for real industrial technology.
Good, very cool. Should we open up this, any questions from the audience? Yeah. Be aware, whatever you want.
Hi. I think we've talked a lot about the kind of output and the analysis and the struggles with analysis, but as Dr. Slagboom had mentioned several times, that the organization, the input of data, what type of tissues, how are they collected, because unless you have the best case scenario, like you described at Providence, where you actually track people before anything happens and you take sequential samples, etc., then how do you make sense of the data in the end? Because it's not controlled at all how the clinical samples have been collected. And I think that might be an issue with, we have problems with reproducibility, because basically you would like sequential samples. You can't always take them in tissue. Are there circulating tumor DNA? Is that something that is under development to enable this sequential tracking and standardization of collections?
Yeah, so so the first thing, we should do is, for example, if you go to a hospital, we have very good eye clinics. Then what they usually do is that you come into a room, as if you go to the bakeries, you get a number, and then you go into a room and they do all kinds of standardized measurements, and then you go to the specialist. And this never happens in a hospital, because you have all these different departments. So whereas in epidemiological studies everybody goes again to the same room being standardly measured, in a real hospital it never happens. So that is the first thing that can be done, is to streamline the first measurement of everybody that comes in, right? And then the next thing is that all the material that has been collected and recorded, for example, we have the largest banks in the Netherlands, tissue banks, are actually from all the tumor samples, and they have been all registered, and now that whole collection has been made ready for use. So now if people want to do, let's say, large-scale RNA sequencing on all those blocks of tumors, then that is linked to those data. And I think, as more and more clinicians see the beauty of the personalized medicine on one hand, the hospitals will start collecting that material in a more rigorous fashion. And the other part is actually what I mentioned, the physiology. So all the challenge tests that you do to people, that really needs to be recorded and to figure out what kind of tissues people are taking. So for example, in all the epidemiological studies, they do OGTT tests, but they never keep the different samples. It's just one measure, and then they throw away all the material, right? So there's a lot that can be done more to fuel these databases with all the time the same kind of data.
Bill, I think that's exactly right. I think one of the challenges that we'll have as we start to see more marrying of electronic health records with genome data, with imaging data, with various, other blood biochemistry. I think managing all of that is going to require a very different type of scientist. So if you're thinking about what careers to do, I think Linda's comment around marrying biology, biochemistry, physiology with data, being able to manage data and interrogate data is going to be, that's what we're struggling with, is finding enough scientists that can do that. And it's going to get exponentially worse. Leroy, we're talking now, I personally talk to countries about how they could start integrating genomic data with all of their electronic health records and all the biochemistry records, and do that prospectively. So you can start to track disease, progression, prevention, and all those things. And we're going to be living in a world where that's happening. And actually it leads to another question, which is ultimately, who are the healthcare companies of the future going to be? Is it going to be the Googles and the Apples and the Amazons who really have a handle on how to handle disparate data sets and actually get information from them? Or, is traditional pharma dead and buried? Where do you see the future being in terms of how we're going to deal with and who's going to be able to manage and capture this data and make it useful?
Well, I have strong feelings about that. I think most of the IT companies, the Microsofts, the Googles, the Oracles, and the IBMs, have really failed in healthcare, repeatedly.
Yes.
And they failed for one reason, and I think they still don't understand it. They have all of the computational expertise you could ever want. They don't understand the importance of domain expertise and the ability to bring that into all the computational tools they have. And you can see it from, Microsoft started some big, ambitious healthcare projects that have gone absolutely to nowhere for exactly that reason. Google is failing in some of its biggest projects now because they've really emphasized the computation to exclusiveness. And I think a role for pharma is that it has a lot of the domain expertise they need, and thinking about how to fashion strategic alliances really would be a worthwhile thing to do. But I think until they can bring in that domain expertise, I don't think they're going to make much of a contribution.
Other thoughts? Where you see devices, wearables, and all these things taking us and just managing big data and what, Google are spinning out now healthcare companies like Calico with actually people with domain expertise. They're trying to do it better than they have done, but it's still a big challenge.
I think the pharma may have to, I agree with Leroy about the tech companies, but I also think that the pharma have to adapt to a very different world, and they're very slow. And for example, aging is a great example, right? So we've been talking about it for 30 years, and maybe few companies now are beginning to think about it, but in a very dismissive way, if you will, right? It's like, maybe one day we'll think about it. And I think they could get surprised, as Microsoft was surprised, and all these companies, were surprised when the new revolutions came about, and some of these big companies, of course, disappeared, and the new companies now, like biotech, right? So Genentech and Amgen, and in the beginning were not taken very seriously, and Leroy was part of that world, and but then of course that became a reality, and I think there's going to be new reality, and certainly longevity, health span, it's going to be a very central reality, and it's just, to me, it's very interesting that there is very, very little understanding and expertise in the big pharma about this, which is going to obviously change the way we do medicine.
It's interesting because there is expertise in the part, things like autophagy and understanding sort of starvation and caloric restriction. I think there is expertise in that and how you can apply that to, for example, cancer. There isn't so much expertise in terms of how you apply it to aging. Even in Parkinson's disease
Well,
and neurodegenerative
even
disease.
cancer, I will argue
Yeah.
there is very little expertise in big pharma about how to use diet in
Not for
cancer
diet, no,
treatment.
no.
Maybe
Talking about the pathway.
supplementation, right?
Talking about the pathways rather than the supplements. I'm talking about understanding, let's say, mTOR, AKT, PI3 kinase pathways.
Sure.
Because
But that's very different from
Yeah,
what
yeah.
I'm talking. I'm talking about what this conference is about,
Yeah,
right?
yeah.
So how do you use, do you really want to go prevent cancer and maybe increase cardiovascular disease or Alzheimer? There are some papers that suggest one as one or the other. That's just very, very bad approach, right? So eventually people are going to figure it out, kind of like what's happening with electric cars, right? And they were kept at bay for a long time, but eventually, I don't know if it's going to happen, but certainly in California, you're starting to see a lot of electric cars around. It makes sense, right?
Oh,
So at some point, I think the longevity intervention is going to make 10 times more sense than most of other interventions.
I think you're right. I think the thing that's going to come before that, though, just again, because we talk about this quite a lot, I think our industry is one of the last to be digitized. And I do think digitization of our industry in terms of how we handle data in itself is the first evolution or revolution that we have to deal with as an industry and in academia and the life sciences as well, because there's just going to be so much data around that if we know how to manipulate, interrogate, and ask questions, I think it will help us pick better targets and ultimately understand aging in a lot more sophisticated way than we do today. Yeah. You're not allowed to do that.
Sorry, yes, I want to ask a question. Because if we say that affecting lifespan can have a general benefit to several diseases, many diseases have been investigated as a separate unit.
Yes.
So there must be drugs around that have benefits for heart disease or for cancer or for other diseases that already may have that cross benefit, right?
Yeah. Where have I got? So up on the third on the left, I've got something called Open Innovation up there, which is one of the things that we're very proud of. So just to give you an example. So exactly for the reason that you're articulating, which is I think we have many very useful tools in our company that may be applicable to things that actually we're not working on or potentially don't have as a high priority as other areas. So we work in AstraZeneca in oncology, in diabetes, in cardiovascular disease, respiratory illness. Those are our main areas. So aging probably for me is still a little bit too nebulous, not because it's not important, I'm just not sure actually how I would go about doing it. Not that it's not interesting, it's very interesting. And all the things I heard today have only got me more excited about the progress that you're making. I'm just not sure if I start an aging program, what study would I do? That's the problem. And if it's prevention, I'm not going to be alive by the time we finished it. And so that's the challenge. How do you do something which is a prevention aging study in a way that actually manifests itself to an output in something that doesn't take 30 years? That's one of the challenges. But to the question that you're asking, we have, when we started this with the Medical Research Council, of which I sit on, we made 22 of our clinical compounds available to every academic in the UK to do whatever they want with. And actually, as a consequence of that, NCATS, which is funded by the NIH, also stood up, and actually there's a number of companies participating in that, where they make clinical compounds available across a number of different mechanisms, pick your favorite one, and NIH investigators can go and tap into them and do either preclinical or clinical experiments with NCATS. They're all clinical experiments. So to give you an example, we have a drug that was in development for oncology, a SRC-Fin kinase inhibitor, that's being developed now by Steve Stripmatter at Yale for Alzheimer's disease. We have a drug that was in development for esophageal reflux, which is being developed now for chronic cough. So all of those things, you can go onto our Open Innovation website and you can access any of our clinical or preclinical compounds. And I think what you see is our industry doing much more of that, where we actually make our know-how and our expertise available to try and help accelerate areas that we're not the areas that we're just interested in, but areas that actually the world and the world is interested in, because we think it will have a benefit. Yeah.
So I would say there are two strategies that pharma is really going to have to change on. So the first, my thesis for a long time has been, You should never treat cancer with a single drug.
Absolutely.
It's a great way to get resistance, and you're
Absolutely.
further behind. We should be giving triple drug or quadruple drug therapy from day one. So what this is going to demand is, how do you do combinatorial triple drug therapy? And I think the key is really going to be microfluidics and single cell analysis, but we can talk about that later. But I would extend...
We're actually doing exactly that in Boston with American Voice. I agree exactly with you.
Yeah. And I would say in most diseases, if you look at the disease-perturbed networks, you realize single drugs by themselves are never going to work. I think most diseases are going to be triple drugs. So it's a whole new strategy that you'll have to think about putting these things together. And actually, I'll disagree with you on the prevention taking 30 years. I would argue if we take large populations, and we hope to have 100,000 patients within the next 2 years on this scientific wellness initiative, they'll give us the earliest transitions for all the common diseases, and they can be picked off one at a time, both to see what the earliest events are and to create the diagnostics and the reversal therapeutics. So I'd argue that's the second area where big pharma ought to invest some money. And I would guess within a 5-year period, some of the easiest diseases will have really figured out how to reverse, and some of them will take a long time.
Yeah, I think that's
wouldn't
reasonable.
argue that
I think that's
point.
reasonable.
So.
And things like, just to give you examples, circulating tumor DNA, which I think someone in the audience asked about, is something that has actually transformed in some tumors at least of how we detect both treatment and the evolution of resistance way before you can see it on a CT scan. And we need more of that. So, and it needs, and I think that actually needs to be not pharma, it needs to be pharma academia together with health authorities, because it requires very large patient numbers that I think any one organization can't do on its own very easily. Yeah.
But sorry, another thing related to that is being ignored though is when you do combination therapy is the toxicity, right? It's completely ignored. So now you have 3 drugs and they're gonna do triple or 10 times the damage, right? And there is zero effort by few companies like, Neulasta, Amgen doing some drugs for the side effects, but minimal effort to look at how do you protect the patient. They might now be on the drug for 10 years against those 3 drugs, right?
So that's why the caloric restriction was, I found that very interesting as a potential approach, and I'll take that back and see whether we have any data on that. Again, I think understanding safety is very important. I completely agree with Reloy that everything about biology tells us 3 or 4 nodes in the pathway to try and avoid resistance, whether you look at antiviral, antibacterial, or I think cancer therapy. The challenge then becomes, there was a question about price earlier. How do you price that, particularly when it's from different companies? Because then you have potentially 3 personalized, very expensive therapies. But more importantly, to your point, the toxicity. And there, I think it's very much about how much you play early with dosing regimen and dose. Because I think also the earlier you are in the disease process, hopefully the lower the dose you need, because actually the disease isn't as progressed. So these are all fantastic points and part of the challenge, I think, exciting challenge that we all face as we go into hopefully more preventive or curative therapies. Just to put it into context for you, to highlight some of the progress that we've made. Metastatic melanoma, 5 years ago, was a death sentence. People, had about 3 to 6 months to live if you were diagnosed with metastatic melanoma. Today, we have patients that are cured with single or monotherapy, or dual therapy, with anti-PD-1, PD-L1s, or anti-PD-L1s combined with CTLA-4s. Harnessing the immune system to tackle oncology is transforming how we treat oncology. Adding on top of that now, targeted kinase therapies, mTOR inhibitors, takes us to another level, but also another level of toxicity. All of those experiments are happening, but they're not easy. And of course, the question is, how do we detect tumors before they develop resistance, right at the beginning of the resistance, so you can get at them very, very early. But all absolutely appropriate points. Let's have another question from the audience. Yeah, right at the back there. Get a microphone to the lady in the back, or you can shout. I'll repeat it. Yeah, microbiome, yep. Let's go to the smart people in the panel first. So, microbiome and aging. Thoughts? Who wants to have a crack at it first?
So I think there is a lot of great animal studies out there on the microbiome. For example, it has been shown that if you introduce a microbiota into the gut of mice that produce more spermidine, these mice would live longer. However, it's terrible tough, much tougher than people think, to change the microbiome of humans. It turns out to be very, very stable, unless you do really some more or less harsh interventions like stool transplantation and these kind of things. So it will not be as easy as just eating a modified yogurt or something like that, because then you have a benefit of 1 or 2 weeks and then your microbiome changes back to the status before.
Other thoughts on how we go about using the microbiome to help us with our aging research?
Well, I'll tell you, we've done microbiome on all the wellness subjects that we've looked at to date. And again, it's mostly discovery. But one absolutely fascinating correlation was a negative correlation between a clinical chemistry result, bile acid, and a particular microbe in the gut. And it was a negative correlation. So we went to the literature and saw, indeed, this microbe did decompose bile acid very beautifully. And there's a disease in the 3rd trimester of pregnancy called cholestasis, where the female tract plugs up, bile acid spills into the blood. People have an enormously uncomfortable 3rd trimester. It becomes acidic. They don't eat because they don't like the food. A really attractive idea would be to make a biotic that could go into the gut with this microbe and manage the bile acid present in the 3rd term. So those are the kind of correlations that are beginning to suggest there'll be really fundamental clinical opportunities there. So I think there are going to be thousands of them. And again, the most elegant studies by far have been done in mice. Not very many things have been done in humans yet.
Question?
When you were in a smaller group, you had this question, if health have anything to do with sleep, and your answer to that was something like it's overrated, sleep is overrated. And I was wondering how many hours do you sleep at night every day?
Tough country. Tough question.
So I usually sleep 5 to 6 hours a night. So I'm on the lower end, I guess. But I haven't had any problems. So.
But I should point out, because I don't think we're going to give the message that the sleep is overrated. I mean, most epidemiological studies suggest that 7 or more hours of sleep are associated with longer lifespan, right? Yeah. There's several of those studies, and some of them are quite large, right? So it doesn't mean that somebody could sleep 2 hours and not do very well. But in general, the 7 to 8 hours of sleep is what is associated with the longer and happier lifespan.
So I heard also during the discussion there was no experimental evidence of benefits of sleep, but actually there are studies in mice, and I don't remember who did these, but they have Alzheimer mice, and what they see during sleep is that they are better able to remove amyloid by opening up the fluid, by letting the fluid flow in the brain. So at least there, I know that, or there's some evidence that there is some benefit of sleep.
They also have disturbed sleep. A lot of the APP transgene mice with the sleep waves.
So you also should take into account that, and this has been shown in humans and mice, that sleep deprivation after some time will put you in a state of pre-diabetes. So your sugar and insulin system will be very, very bad shape. Okay?
I highly recommend sleep. But it's only 20 hours of it. And the younger you are, the easier it is as well. Other questions? Any other questions? Yeah. At the back there in the purple sweater. Can we get the microphone back there? Oh, go and ask here, then we'll go to the back afterwards. Go on. Ask at the front here.
Oh,
yeah.
Okay. Yeah, I was wondering, there are cells called HeLa cells, which are human cancer cells that were extracted really long time ago and are immortal, as I've understood it. And they're used in research in many other areas, but I don't really know if they're used in aging research or not, but it feels like a relevant
Which cells?
part.
What are they called?
HeLa cells.
HeLa cells.
And I was wondering on your opinions, or if you know any research about it and so on.
Thomas, do you want to talk about HeLa cells?
Not really. I think it's been mentioned here before that a lot of these cell lines turn out not to be so relevant when it comes to aging research because they've been immortalized and genetically and so on and so forth. So it's hard to squeak out any normal process that would go on during aging. With HeLa cells is just one example. There are other examples of cancer cell lines as well that is probably not so informative.
Yeah. I think if I could summarize what I've heard the panel say and something that we very much agree with, I think we're moving away from transformed cell lines, even cancer cell lines in our oncology studies. We're moving more to patient-derived tumor samples or induced pluripotent stem cell complex models, 3D models, either on chips or in complex organoid cultures, because we find they're much more reflective of the human organs than anything we can do with a transformed cell line. I think that's the way generally I think science is evolving that way, which I think is what you've heard the panels say. There was a question at the back, yeah.
I'm not sure. Yeah, I think it was Dr. Leroy Hood who spoke of how treating cancer with a single drug is a great way of building up resistance. And it made me think about antibiotics resistance. I have a couple of questions about that. Just general thoughts, and I wonder, is it inevitable? And what then, when it comes? Is human life expectancy going to decrease?
Well, I would say to a first approximation, treating cancer with a single drug is generally inevitable in creating resistance. There are some exceptions, and actually some of the spectacular exceptions are in using immunotherapy for certain kinds of drugs like melanoma. That by itself has succeeded in curing. But I think the genetic polymorphism in the human population, I don't think we're going to find single drugs that universally can cure any particular type of cancer. I guess the question of if we can cure chronic diseases that are major in nature, are we going to increase the aged population because these people haven't died off in an appropriate way? I think with scientific wellness, we're enormously going to shift the average age of the population to older individuals, but the older individuals will be very functional and will have accumulated a great deal of wisdom. But what society is going to have to rethink is what retirement means and how we can ensure young people can have jobs in the future. And those are both non-trivial problems to deal with.
In the front, yep.
Okay. It was mentioned earlier that administering biotics could be quite complicated into the guts. What then is the general consensus on yogurts with lactic acid bacteria that, as far as I know, have been proven to change the microflora? Yeah.
So you're saying how does lactic
the flora,
acidosis change the microbiotic flora?
How do you change it?
No, I think it's how does in jog you're asking about.
It was mentioned that it is complicated to to change the flora.
Right.
But then, are these advertisements totally false, or is there some scientific nature behind them at all?
I think the question is, the comment was it's complicated to change the microbiome, but some studies have been done that suggest that you can do it. Are they incorrect or?
Well, there's one great way that you can fundamentally change your microbiome, and that's take broad-spectrum antibiotics.
Yeah.
And it takes far more than a year for your microbiome to even begin to recover after those broad-spectrum antibiotics. And I think in the future what we'll do is have your microbiome in a bottle, so when you finish your broad-spectrum antibiotics, they can give it back to you and reconstitute them. But I think in humans, there's very little experimentation been done on how to change the constitution of the microbiome effectively. There are a lot of prebiotics and probiotics, but I don't think very much science has been done on any of those things.
Well, there's a number of papers. For example, one that was published in Nature a few years ago looked at the switch to a vegan diet for five days, I think it was, and that had a major impact on the microbiome. That was also long-lasting. Now, how long-lasting, they didn't determine it, but I would say that the intervening with the probiota only may be a little bit simplistic. So you have a problem, you have a gut that is full of a certain profile of bacteria, and just going in with a different kind may be simplistic, because you have to replace the bad one, as was just mentioned, and then you have to find a way for that to also cooperate with the rest, right? And so it's much more complicated than I think some of these companies that you mentioned want you to think. But some of it might work. I think a more comprehensive intervention that's smarter, and that, like the one I just mentioned in that paper, they can replace in a coordinated way the old bacteria with new bacteria, because now the new bacteria are eating up on the new diet, right? And that's what. And I think there was also a paper showing that a primate, monkey, and human microbiota was more similar if they were vegetarian than human and human. They were carnivores or vegetarian. I mean, this is how powerful the diet that you have can be on your microbiota.
Frank, you have a comment? Yep.
So this goes in line with another paper showing that even with mice, you cannot change their microbiome just by oral intake of microbiota. Only if you, for a long, long time, supplement them with plant fibers. So plant fibers together with new microbiota would change then the microbiome finally. So it's not as easy as having a drink. So you also have to nurture the bugs you take, in order to get them established.
In the work that I'm aware of is fecal transplant work, where patients with C. diff infections and very severe IBS have had fecal transplants, and that has had quite dramatic effects. There was a small company in Boston that tried to distill out the positive factors in the feces and kind of create a pill, but that trial actually just read out a few weeks ago and failed quite dramatically. So it's not easy to take the complexity of the microbiome and recapitulate it in a pill, unfortunately. There was another question, I think I saw a hand up, whoever's got the microphone, ask your question.
My question was...
With the microphone though.
Is it on? Yeah, it is. So my question was, if we're now living in a much cleaner environment than before, does that in any way affect our susceptibility to age-related diseases in any way? Do you understand my question?
Yep. So basically, with the increase in hygiene and everyone washing their hands and being careful, what does that do to our resistance to to bacteria and infections? I think that's your question. Yep.
Well, I think the most classic example of that being deleterious is when you go from the nice, clean environment to a lot of third world countries, you are incredibly susceptible to microbial gut infections, a whole variety of different kinds of things. So I think our clean environment does make us significantly less immunologically responsive to a diverse range of pathogens and things.
Linda, yep.
I think it's also very important with lab animals. So there's been some quite interesting publications recently about mice, because one of the things that we all have to do if we keep mice and are doing aging research is to keep them specific pathogen free, and we have to prove that they're free of those specific pathogens so that there isn't a complication from various infections in the results. But what that actually means is that the immune system of the mice doesn't develop normally. And people who keep their mice in a slightly dirty house often get completely different results with metabolic interventions from people working with these terribly clean mice. And it's really quite a source of tension with the authorities and with the veterinarians who decide on the standards in a particular mouse facility, just how dirty it should be in order to be realistic. So there's a real issue there. It's a very important question.
Great point. Let me ask you one last question.
Could I just extend my question to the age-related diseases of humans contra the clean environment? Do you understand? Like Alzheimer's, Parkinson's.
Does the clean environment change how we age, or our healthy
them?
aging? I think that's the question.
I can't answer that.
Unknown, I think, is the answer. Let me just ask you a question.
Obviously, the clean environment is the major factor that has extended lifespan in the past 200 years, right?
Yeah.
So that's for sure the positive side of it. But now, could it have now a negative impact by making us too sensitive to future stresses?
The title of the conference is around model organisms to humans, and you've all presented, I think, a lot of work, both in the preclinical and some of you in the clinical setting. One of the questions I often get asked is, should we be doing any experiments in animals and preclinical organisms, or should we be doing everything in humans or in cell-based systems? Particularly given the audience that we have, the young audience, maybe a few of you could comment on the importance, right or wrong, of whether we should be doing experimenting model organisms, animals, the benefit of doing those experiments versus not.
To kick off in favour of using the organisms, obviously you should use the minimum type of animal and the type of intervention that's going to answer your question adequately. But aging is such a whole organism phenomenon with the different parts of the body talking to each other and defects in one tissue knocking onto things going wrong in another. I think it would be almost impossible to get the answers that we need if we didn't look at organisms.
And maybe I want to add what I already wanted to say earlier. So being involved in osteoarthritis research has been extremely important to make the right animal model. So for example, animal models for osteoarthritis that have been used is where they cut the ligaments and then the animal develops osteoarthritis in a week, which is actually a highly inflammatory process, and that is not the osteoarthritis that a 60-year-old develops, right? So it's extremely important that the right animal models are developed for the human situation. But having said that, if such an animal model is there, it's also extremely important that if medication is then developed, that it's also given to the older animal, right? And that is frequently not done. So the medication is developed, and when it's tested for toxicity or the right doses, or it's always in the younger animals, it's rarely in the older animals. And so I think that we could use even the animal models better than we do.
It's even worse than that, actually. It's not even that we don't do, well, again, we try and do, but many don't do.
Yeah.
It's not just even doing it in older animals, it's actually doing it in older sick animals. Yeah. So if you're trying to treat a cardiovascular patient, what you really want to do your toxicity experiments in is in an animal that has CV liabilities or toxicities, so that you can see any exacerbation of the fact that you're actually treating someone that's not well versus someone that's healthy. So it's a great point. The challenge is quite often rodents, dogs, rabbits don't reflect human toxicity very well, which is why moving to human-derived models, I think, particularly organoids, is quite often a very good step before you go into the toxicology experiments in animals. I just want to take a moment to thank the panel. I mean, I've been here today. I wasn't here yesterday, unfortunately, but it was absolutely fantastic set of presentations, very inspiring, hopefully motivating to all of you. If you're thinking about a scientific career, go for it, because there's nothing better. than having a really cool scientific career. I think you get a sense from everyone here how much they love their jobs, their roles, the adventures they have, the debates they have, and that's very much true for all of our science work in AstraZeneca as well. There's, to me, no better career than a really cool scientific career. So I hope you've had a really, really good couple of days, and I'd like to thank all of our panel for a great session.
So now, everybody, it's time for me to close this very interesting Molecular Frontiers Symposium. I had in mind to summarize what we have learned and also ask some questions. But now, having understood that the Leroy 5 hours of sleep is not enough, and I see that you are quite tired, quite a few of you, me too, actually. I will not do that, but I think we might type these questions, and you could take them as homework, and something that could qualify you for the next Molecular Frontiers Symposium, which will be 22nd to 24th of May next year. And the title of that symposium is something like Man-Tailored Biology, from the fundamentals to medicine. So it's again a topic that is close to medicine and biology. That's not always the case. Typical for Molecular Frontiers is that we use the molecule as the vehicle to enter some interesting problem. It could be energy, it could be environment, and so on. But I think we should thank all the speakers for terrific talks. It has been really fantastic to follow it, and I've noticed that many of you have enjoyed it as much as I have. I would also like to thank you for coming up with so good questions, and I'm sure we will continue to have contact and communicate about Molecular Frontiers. And finally, I want to thank those who have done the job here, organized the conference, and that's Dina. Could you stand up again? And it's Per Thorén. Yes. And it's Åsa Rensfeldt. Okay. So with that, I close this symposium.